So for folks that just joined in, we're up in the water column about 60 meters above the bottom. We're just getting the larval pump on board that we released, and then we're going to get set up to do a transit up to a site called Zeta Garden, which is a low temperature, very iron-rich bacterial mat site that we're going to look at briefly, and we're taking a sample for Craig Moyer, a colleague who has been working here at the site, and then we're going to be investigating Q vent, which is just a little north of that, but the main focus of this dive is sampling M vent and bio vent, which are up around 951 north. So in just hopefully not too long, few minutes, 10 minutes, we're going to get back down to the seafloor, and we're going to be traversing over a very large lava channel that was part of the distribution network for the 2005-2006 flow on the west side of the axis that transported lava out across the west side of the EPR crestal plateau here.
Hi then. On the dive plan, I saw that.
Hi, Nuno.
Hey, I saw on point 11 that you were planning to do image at the M-vent instrument site. Is that acquiring images for doing 3D mapping there, or?
Well, we hope so. Seeing as how I have you and Javier on the line, I have been so busy that I haven't had a chance to write you a memo, but I was planning on doing that. What's become obvious to me is we, I think, have areas where we can do successful 3D imaging, but in some areas, the sites are complicated, because of a lot of diffuse flow coming out of the chimneys, and in addition, there are complications with respect to adjacent structures. So what I'm hoping to do is, based on the work that we've done at Bio9 and PVent, as well as what we hope to do today at M and BioVent, is to try to see what areas might be most productive for where we can do the quantitative 3D imaging. We'll try to do some of that, and I can hopefully get you snippets of what we've been doing, or you can see what we've been doing. But my plan, if things go well, is that we would then devote a dive or two specifically to the imaging, seeing as how we will have accomplished the fluid sampling and the instrumentation with event loggers. So I think what we're doing now is a bit of reconnaissance to see how well we might be able to do the imaging, which of the vent structures might be most amenable to doing that. And so that's sort of where my thinking is right now about it. So I think we'll try to do some, but the focus for this dive is to try to make sure that we're accomplishing the fluid sampling and the instrumentation, putting the loggers in the vents, but with also certainly a partial focus on collecting some really good imagery. Whether it's what you'll need for your quantitative work is not totally clear, and again, I think we need to look at it a bit more carefully. So your joining in this evening would be or tomorrow morning would be great. That work is likely to happen, over the course of probably the next 6-8 hours or something like that. We've got about an 18-hour dive, 18-hour dive.
Okay, yeah, exactly. That's exactly the point that I was. If you had any idea more or less when that would take place, of course, it's very hard to tell. But okay, but it will not be in the next couple of next few hours. Okay. I'll try to join in. Now it's pretty late over here, but I will try to join in the morning when I get that.
Oh, okay. Sounds good, Nuno. Thank you. Okay. Yeah, we'll certainly be having more discussions about this.
Okay, perfect.
I'm just going to break off for a second to make sure that we get on track and headed out. Right now, we're just about a couple of 100 meters west of Bio9 vent and P vent, and we're going to be traversing the seafloor soon. We're going to get back down to the bottom. Right now, we're about 55 meters above the bottom. So we've just got a watch change going on, so there's a lot of extraneous noise in here. We're going to be getting going in just a little bit. All right, are we almost sorted out here so we can get going? Yeah, so we're getting down to the bottom. We're at right now 44. He's coming down, and we're going to be doing a traverse up the west side of the axis, just outside the axis of the axial trough, over a big lava channel to start with, and then just over the collapsed terrain, the lobate terrain, right along the west margin of the AST. I'm doing, yeah.
Hey, Dan.
Yes, Javier, go ahead.
Yeah, I have a question. Is there navigation files or a map update or something of what has been mapped with Sentry and the tracks of Jason? Or
We have renav for all of the dives up to yesterday's. It takes them about 3 or 4 hours to do the renavigation. That's the integration of the USBL with the DVL Doppler, bottom lock nav. So we do have all of those. So in terms of being able to provide, navigation files for Jason for the surveys that we've done with a downlooking camera, we do have that.
Okay. Can you share at some point some maps to know where we are with respect to what was planned? Because
yes, I will talk to Jandai about both providing some maps and identifying where on those dive tracks we did specific sort of down-looking imaging. Sometimes we were doing a transit. Sometimes it was on purpose that we wanted to sort of cover that terrain, but we'll definitely do that.
Okay.
To be honest, it's the old adage of using an ROV and drinking from the fire hose. We've been keeping up, but it's a little challenging, let's say.
Yeah, of course.
But the team out here has been doing a great job.
And in terms of image quality, is it good, Sentry?
The image quality is great, especially so. Okay. Hang on a sec, Akel.
Yeah.
Excuse me. We want to have the basket out for the strobe. You want to go right to that cursor. That's what we want to ship, because that'll allow us to work at Zeta Garden up to the northwest of it. That's fine. Yeah. Is that out as far as it can go? There you go. Okay. Thank you. Right. We just put the basket out because I've mounted one strobe on the basket. That distribution of the lighting, Nuno and Javier, has helped a lot in terms of making the lighting from the Jason down looking a lot more even. Okay, so we going? Great. Okay, and if you can, yeah, 6 meters is great. Akel, thank you. And you on altimeter for how you're controlling the altitude lock? Yeah, auto alt would be best. Thank you. Okay. All right, let's get going. So we're going to be traversing, you see these low bait sort of tubular lavas here right now. Our heading is just about due north, 1 degree. So we're going to be on this course heading directly up along the west side of the axial trough.
Altitude.
Who's ever talking, can you mute your microphone, please? Javier, I think that's your iPod.
It was you on YouTube. Sorry.
Oh, okay. So is the YouTube channel that Ross set up working?
Yeah, I just it's by mistake I turned on the volume of my computer and it was
no worries. But
it's working. It's working.
We'll be interested to see, because we did send that to several other people as well.
Yeah, that's working well. Great.
Let me see who else is on there. Just for a second. Just here. Yes. Okay, that's great. Okay, great. Okay, so yeah, so it shows that we're live on YouTube. That's great. So I just want to briefly mention a couple of things. So one of the colleagues that's on the call here, Rachel Heyman, she and I have been working here for a long time, over 30 years. Initially discovered these sites that we're working at in 1989. Hi, Rachel. Glad that you could join. I don't know if both you and Ken are on the line or watching, but thrilled that you could join us. And Tim, I'm glad you got the message too, and can join in. We'll be doing some things probably of interest to you in a while, but that's great. And then the other person who's on, a longtime colleague of mine, Jim Casalos, who helped to develop one of the first mapping systems that was used to map these Pacific Rise, the original CMark systems. He's logged in as well. Jim, I hope you and Deb are doing well. So yeah, and I think the challenge for you is going to be just making sure you keep at least the visual that they've got, which is a science cam, at least, yeah, clear as it can be, right? And just so we don't forget, we do, once we get set up at Zeta Garden, we'll want a white balance. And I made a white part to the white balance thing, the target. So we're at about 5 meters altitude right now. Pretty good view. You're looking at in the Zoom screen and the YouTube screen. You're looking at the Sulis 4K video camera that's been downgraded to a 1080p high definition image, and that's being controlled largely by the watch leader. Okay. And now it's okay. All right. Can you open up the ISO a little bit more? And I don't know, I have no idea where the iris is set right now, so you just might check on that. Did
done?
Wait 1 second, please. Yes, Javier.
How far are you from the target?
Just hang on a second. We're trying to get some video stuff sorted out.
Oh, sorry.
We're about 1 kilometer, maybe a little bit less.
Sorry, I missed that.
We're about 1 kilometer.
Roger.
So what happened? It's going to take us about an hour to get up there.
So there's just not, there isn't enough light for the
No, with you, the camera is on. I can see the camera back here. It's just a question of how it's being routed, I think. So we've just entered into the lava channel that's just west of the axis. This was part of the distribution network for the for some of the 2005-06 lava flows. Lots of curtain folded and sort of hackly lava. And we're going across the direction of travel. So you see all of those folds are going from the right-hand side of the screen to the left-hand side of the screen. That was the pretty fast moving lava flow that was coming out from the eruptive source, probably fissures right in the axial trough. We got it, Chris, or James got it. Sorry. Can you tell me what the range is from us to Zeta Garden is? You got to keep going. You got to, yeah, no, reduce the map. Zoom out on the map.
There it is.
Yeah.
So Zeta Garden is just over 500.
Okay, 500 meters away.
Yeah.
2, 3. Okay. So we're about a little more than 500 meters away from the Zeta Garden site. And this channel here has several levees. So here you're seeing a transition from the hackly jumbled lavas in the foreground down to the lower left to more sheet flows in the upper right. Thanks, Akel. This is a good view both for the video and for the downlooking. This is great in terms of altitude. Yeah, that's okay. Are you getting tugged by the cable? Yeah. Yeah, that's fine. Hi, CL. Glad you got the message.
Hi, yep, just got off a clubhouse room, so.
Oh, there you go. From one social media adventure to the next.
Exactly. The marvelous world.
Well, we're just sorted out and heading to this site that we're going to start work at tonight. It's about 500 meters away, so it'll be, unless you're really interested in lava flows, it will be more exciting with opportunistic and charismatic megafauna coming up soon.
All right, I'll dive in and out.
Okay. All right. Thanks. Did you send a message to Simon so that he would know about it too?
I'm about to send him a text.
Okay, great. Thanks, sweetie.
Hey, Dan, it's Tim.
Hey, Tim. Hi.
Yeah.
Glad you could join in.
Me too. And I'm don't want to interrupt the video for long. I said a quick question is, are you traversing sort of, what's your heading? I'm trying to figure out, are you going along a wall?
North. Tim,
Straight down the middle.
We're heading north. No, we're not in the axis. We're west of the axis in a big lava channel that's part of the 0506 lava flow system, and a major channel that distributed lava out to the west and northwest. So we're about 150 meters west of the west AST wall. We're just a little north of the latitude of Tika, and heading straight for Zeta Garden.
Perfect. Thank you.
Okay. Yeah, I'm sorry that I can't somehow broadcast the map. Actually, hold on. Let me ask Akel this question. Akel, is there any chance that I can change from the 4K to that without having to rummage around in the video box to show that? Who you can do it, Nicole. So hang on a second. Hold on. This is an adventure in video controls here. We're looking at the map that was made with the Sentry in 2018, 2019, a 1 meter resolution bathymetric map. I want to send the NavG screen. It's not on there. Hello, Kevin. Thanks for joining. Kevin Johnson from NSF just joined in, the program manager who's helping to support the geology program here during this cruise.
Yep. Thanks, Dan. Good to see
Okay,
you.
So here you're looking at the Sentry bathymetric map, and that's what we're using to fly the ROV. I'm assuming everybody can see that that bathymetric map. You're looking at that oblong rectangle, that's the ship, and down at the bottom is a little yellow rectangle, which is the ROV, and the axial trough is that sort of greenish colored topography in about the middle of the map, and that's the axial trough right there. We're heading up to a site called Zeta Garden, which is up at the top of the map in the middle, that little X up on the top. I'm assuming folks can see that map. Yes, great. Thanks, Kevin, for the thumbs up. Thank you. Yeah, we just spent 5 minutes figuring out how to route the right video channel to the screen. So that gives everybody a bit of a sense of where we're at. The map that you're looking at was made by Jun-Nai Wu and Ross Parnell-Turner with help from Greg Curris at Seafloor Investigations during using data that we collected on Atlantis cruises here in 2018 and 2019, also supported by NSF and Kevin's program in the MG&G program. Okay, so now that everybody's more or less oriented, and do you understand, Nicole, how to switch it back? That would be great. So let's switch it back to the video. Okay, look at that. Amazing. All right, perfect. Good job. We're just trying to focus the camera here while we're traversing to get the focus good, and then we'll zoom back out. Yeah, okay, that's fine. It's close, yeah. Let's zoom out. Yeah.
That was Justin. He said that Sentry, they're setting up for 7 o'clock recovery with
Yeah. What time is it now?
So
Wait. Oh, hold on a second.
4:15 now.
I got plenty of time. So,
This is just
yeah. Well, we're going to be in the right place, and so we don't have to leave when they pop Sentry. Okay, so we're fine right now. So just so you know, part of the complexity of this particular field program is coordinating the operations of the Sentry AUV, also part of WHOI's NDSF. Sentry is actually in front of us and to the west, mapping, doing a detailed photo survey using a down-looking camera. And it's going to be doing that survey for the next several hours, and then it'll be recovered. So we'll break off of our work, and we will then go and recover Sentry, and then continue with the rest of the work into the wee hours and into tomorrow morning. You see that plate that the pilot is ditching. He's ballasting the ROV, so it's has the proper buoyancy, not too heavy, not too light. Sort of like morning coffee, and so we're just doing that for a moment, and that makes operations when we're doing sampling much more efficient, so that he's not having to drive down too hard and stir up sediment and whatnot as we're doing the sampling operations. Ross, if you're on the call, seeing as how you arrange the YouTube channel, you should let me know if you think I need to provide any more context. Oh, he just stepped away. All right. Okay. All right. Yeah. Okay. So because I'm not very familiar with what actually is happening on YouTube, it's unclear whether I should. I'm assuming my voice is being broadcast. I think it is. Is that true, Nicole?
Then I can confirm that your voice comes out and everything was
on YouTube.
Yeah, so it's great.
Oh, okay. All right. So what I'll do is provide a little context for whoever else on the planet is listening in. We're at about 2,500 meters depth, working with a remotely operated vehicle, Jason, operated by Woods Hole Oceanographic Institution in Cape Cod, Massachusetts. And we also simultaneously have an autonomous underwater vehicle, Sentry, also operated by Woods Hole's National Deep Submergence Facility, supported by the federal agencies, primarily the National Science Foundation. And we're doing research, basic research here, geological and geochemical research, studying seafloor volcanism and hydrothermal processes. We're using the Scripps Institution of Oceanography vessel RV Revelle, the Roger Revelle. And Scripps Institution is a part of the University of California at San Diego. So it's a combination of using a very capable oceanographic ship and capable deep sea vehicles to be able to actually traverse the bottom and sampling and make the observations that we need to study basically the creation of ocean crust, which is where most of the volcanism on Earth occurs, in the bottom of the ocean, hidden from view for the most part, except for you folks looking at this. This is the results of what happens over time. There's lots of basaltic rock that gets erupted on the seafloor at the mid-ocean ridge, which is a globe encircling ridge system that's composed of many volcanoes, volcanic centers. So that's in very brief a sort of summary of what we're doing and how we're doing it. We've got engineers from many different places, institutions, primarily Scripps and Woods Hole Oceanographic, but also others that are helping. And we have science parties from Woods Hole Oceanographic and Scripps Institution of Oceanography, Lehigh University, Oregon State University, Rutgers University. So there's many co-PIs cooperating. There's two science teams, a biology team led by Lauren Molyneux, who's a senior scientist at Woods Hole Oceanographic. And they're looking at larval processes here, how the larvae of these vent animals sort of colonize the various hydrothermal vents. And then a team of scientists, Ross Parnell-Turner at Scripps Institution, Jill McDermott at Lehigh University, Thibaut Barreyre at the University of Bergen in Norway, and myself, I'm Dan Fornari at Woods Hole Oceanographic. And we're collaborating on these studies. It's supported both by the Biological Oceanography Program at the National Science Foundation and the Marine Geology and Geophysics Program at NSF. All right, so that's a little bit of a summary for whoever happened to be listening to that. And we also have some additional collaborators who are working with us. There's Santiago Herrera, a biologist, a deep sea biologist, also from Lehigh University, Nuno Garcia at the University of Girona, and Javier Escartin at CNRS in France. We're traversing over the central part of this lava channel here. As you can see, it's mostly sheet flows, not too many folded flows in this area, but further to the south, where the margin of the flow was, it was a very chaotic terrain of curtain folded and Hackley lava flows. And these lavas were erupted about 15 years ago. One of the focuses of our work is to look at the what we hope will be another eruption here that might occur in a few years. And we're basing that on our monitoring and the vent fluid temperatures and the chemistry of the vent fluids, as well as sort of observations that we're making of the faunal communities around the vents, the biology, which oftentimes responds in pretty profound ways to how much heat is coming out. And the heat here is provided by magma that's about 1.5 kilometers or 1.2 kilometers beneath the seafloor in melt lenses that are being replenished from the mantle. No, stop. Don't.
So, for example,
So here you're seeing a section that is more curtain folded and jumbled. Those big folds are entirely volcanic glass. Those of you that know something about rocks, it's like obsidian. So it's a very quickly chilled volcanic glass or volcanic rock. There's glass crusts here that are as thick as a few centimeters. So, you know, an inch or more. And some of those folds are entirely glass. So that means that as the lava was flowing quite fast, it just quenched, the surface quenched and created this smooth surface. But underneath, the lava kept flowing out to the west, because we know that it reached as much as 3 kilometers to the west of this location where we're at right now. Okay, so how far do we have? We have 200 and I don't know, 200, almost 300 meters, a little less than 300 meters to go. Okay, so we're about 300 meters from the Zeta Garden site, where we'll be stopping to do some work. We're going to be trying to measure fluids. It's a site that has a lot of iron-rich bacterial mats in it, We don't know what the temperatures are. We didn't. We observed that on our first dive when we did a bit of reconnaissance and drove up here. It seemed to have diffuse flow that was flowing out of cracks in the basalt. And again, they're quite extensive bacterial mats, but a lot of iron-rich flock as well. You trying to focus on those? Oh, no sound. I
hear you now.
don't think they got any of this. Hey, can you hear me now?
I can hear
Mike?
you now, but I didn't up to this point.
Okay, yeah, I think I provided that whole dialogue to myself, basically. Sorry, thanks, Mike.
Yeah, all right.
All right, so none of the description of what was on the basket made it through, right? Is that right, Mike?
No, yeah, I didn't hear anything about the basket. Nope.
I didn't hear anything either.
Or anything about the low base or anything like that.
Yeah. Oh, okay. All right. So let me start again. I'm sorry. I was muted. I'm still getting used to this microphone here. So I was saying that I was just giving people a view of the basket and the kind of samplers that we're carrying. The black box on the right-hand side is the bio box. It actually contains not biology for our dive, but 3 temperature loggers made by EP Oceanographic and developed by me and Thibaut to measure the vent fluid temperatures. You can't see them right now because they're hidden in the box. And then in the middle and lower left, there are some major fluid samplers. Those are titanium syringe samplers, and they're used to collect the fluids from the hot vents. The vent temperatures 350 degrees centigrade. These vents that we're working at are in the range of about 360 to 375, maybe a little hotter. That's one of the indicators that we get of maybe how close we are to the next eruption if the vent temperatures are increasing. Once we get to about 390 degrees or the high 80s, it looks like the melt may be shallowing sufficiently, so we're getting those very hot temperatures. So far, the fluids that we've gotten are less than seawater, so they do appear to be vapor phase or phase separating, which is also another good indicator in terms of perhaps the melt coming closer to the seafloor and eventually erupting at some point soon. The last type of sampler we have is those bottles labeled 3 and 4, which are IGTs, isobaric gas tights. Those are developed by Jeff Seawald at Woods Hole, Jill McDermott, who is one of Jeff's PhD students. She's a co-PI on this project, and those samplers are being maintained by her team and sampled at the vents so that we could collect the gases, because the syringe samplers, the major samplers, don't retain the gases. The seals are not designed to be able to do that. So those are the kinds of samplers we have. And then I was also pointing out the camera system. We have a variety of camera systems that were developed using NSF funding, and the ones that you see on the manipulator there were developed by me and my colleague Eli Perrone at Ocean Imaging Systems, and they're GoPro cameras and a deep sea power and light housing, and that little camera on the top is a viewfinder for those cameras, and we're using them to image the vents and the vent surfaces and the volcanic terrain. One of them is taking 12 megapixel stills every 2 seconds, and the other one is collecting 4K video, and they can do that for the entire duration of the dive. About 18 hours. And in addition, we have a down-looking camera. That flash that you're seeing in the bottom of the screen is a 24 megapixel camera, also developed by Eli and Ocean Imaging Systems. And we're using that to collect very high resolution pictures vertically downwards using 2 strobes. All of the light that you see here on the remotely operated vehicle is LED, light emitting diodes, and made by Deep Sea Power and Light, and they have are a leader in the field of deep sea imaging and lighting.
Oh, sounds like you're muted, Dan.
Yes, I'm muting myself sometimes, so when I'm having discussions with the other watchstanders and whatnot. So Mike, we're west of the AST by about a couple of 100 meters, and we're actually we're about out of the lava channel, one of those main lava channels that was northwest of Tika Vent. And we've got Sentry mapping with one of my down-looking cameras, the part of the lava channel off to the west of us. Yeah, it's about the 200 meters west of us, doing a north-south traverse at 5 meters track line spacing at about 6 meters altitude. So we've got the ship almost to where we want to sample. The vehicle is catching up to it. We've got about a little more than 200 meters to go, 240 meters or so. Okay, just so we're getting quite close to our site, about 200 meters, and then it's going to start to get pretty busy. But I just wanted to introduce the Jason team who's currently in the van. We have 10 people from the Jason team at the National Deep Submergence Facility at Hui. Kel Kiva Sterling is the pilot. Hugh Popenoe is the navigator on this watch, and Ben Pietro is the engineer who's manning the controls of the winch and other devices. He's assisting the pilot at the moment. Many of them switch off. The pilot is the lead person on this watch for the NDSF vehicle team, for the Jason team, and the other two people are supporting him in basically driving the vehicle where we want it to go and taking the samples that we need to take and making the observations so that we can make the observations. And then in the back, the science team, there's 4 people in here right now, including myself. So there's Nicole Pator at Lehigh University, and Connor Downing at also at Lehigh, students of Jill McDermott's, and Zoe Yin at Scripps Institution of Oceanography, who is kindly agreed to come out and get some experience going to sea. And me, I'm Dan Fornari from Woods Hole Oceanographic, a marine geologist. It's on the swing arm on the port side. So just so you know, you may want to look away from your screen for a minute, because we're gonna do a white balance, so it's gonna be totally white. We're trying to make sure that the camera is as color balanced as well as it can be. So that's why we're moving things around for a minute, so that we don't have to do that when we get to our sampling site. You see that? No, I guess that basket didn't really come into view. We have 2 swing arms in the side that have bio boxes as well. What's that? Oh, tell me that they didn't put it on. They didn't put it on. Did you check the other one? It was there. I'm wondering whether they didn't put it on. No, on the starboard side. Well, you know, they had another sampler in there on the last dive, and so that got replaced. Maybe we won't do the white balance. No, there isn't one on the elevator. Okay, so no white balance. We don't have the target with us. We forgot it at the surface because we've been switching out between biology and geology dives. So we'll do that next time, and we'll try as much as possible, perhaps, to use some opportunistic tube worms, which are mostly white, and we'll try to zoom in on them to see if we can get a white balance. Okay, we're about 100 meters due south of Zeta Garden. We're just coming over the axial trough wall now. You see that topography is dropping off to the right side of the screen. That's the sort of step in the wall, which can be from 3 meters to maybe 5 or 7 meters high, and that's where most of the volcanic fissures that produce the lava that came out last time are located. And it's where the majority of the hydrothermal vents along this ridge are located. And you'll start to see more sort of animals, crabs and serpulid worms. You'll see what we call the suburbs, if you will, of the vent communities as we start to get closer to the source of the venting in the middle of the axis. We're about 4.5 meters altitude right now. And there's all that jumbled stuff, jumbled lava crusts on the left side of the screen. That's all the collapsed talus. And we'll probably see a bunch of lava pillars that will be along the margin and sometimes along the center of the axial trough here. Are you going to be in a good spot, Akel, in terms of the ship and working there, seeing as how we're going north? Okay. Can you get down a little bit closer to the bottom? Okay. Yeah, so I would take it out of sort of flying mode and let's get down to a couple of meters. And let's turn off my camera. Let's turn off the down camera. So if you could log that, or down camera's off now.
Justin, I'm glad, but it's always good to get the people call.
And can you rotate the port arm back over to in front of us so that I can get that in the view as well? We're just moving the starboard or the port manipulator over that has the camera system on it so that we get a better view of the seafloor in that. That's good. That's good.
I could turn on the beacon so you can see it.
I think that's pretty good. Yeah, you could have focused on that holothurian, but just go to the right and focus on the holothurian. And yeah, that. Okay, zoom in all the way on that. See if you can see the spines on his back, which you can for the most part. That looks pretty good to me. Yeah, that's good. That's good. Yeah, yeah. Go back. Zoom out.
It's not as great as it must look to you, Dan. I don't know why, but sort of fuzzy. The image is sort of jumping around a little bit in spots.
Are you watching on the Zoom link or on the?
No, I'm watching on your link, not on the. Yeah, on the Zoom link, not on the YouTube link.
Yeah. Okay. Well, I mean, this is one of the reasons why we opened it up a bit and had a bunch of people join, is to see whether we're experiencing any impact if lots of people join. Do we know how many people we've got on here? 10. That's not too bad.
Mike, it's not jumping around for me. It's pretty steady for me.
Really?
Yeah.
I think it's your internet down in Florida, Mike.
What else could go wrong? Yeah. No, it wasn't.
Yeah, right.
It wasn't like the whole image was jumping around. It's like it was sort of. It's like you were underwater. Oh, that's funny. Yeah, right. It was just sort of well, it's just not as clear. It was really so clear the other day, and right now it's sort of okay, at least for me here in Florida.
Yeah, right. Okay, so that's Mike Perfit, my longtime colleague in Florida, the professor of geology at the University of Florida. Part of that, Mike, is there's a fair amount of particulate matter in the water here, more than normal or more than we've experienced before, and so that's some of it. But but you know, you'll see here in a minute when we start to work at the site. And actually, we're just coming onto it now. There's some, and this site is it's a little tough to work at because of all of this iron-rich bacterial flock. So we're going to be looking around for that site. There's off to the right, Raquel, right down there. Yeah. Okay, so that's some of it, but there are areas that are quite iron-rich, that sort of brownish sediment that you see. There, he's just panning over to it right now. So this is the Zeta Garden site, and let's pan up to see what that structure is. So, right. Okay, so and then down a little bit and off to the right. So we also have Rose Jones, who's a postdoctoral student of Barreyre at the University of Minnesota, who's been out on several cruises here, working with Jason Sylvan. You have to come down on the ISO, so try to keep that in control. Okay, so I'm just going to talk with Rose a bit here and figure out where we want to be.
If you care, everything also looks, again, to me, looks very green. Green and
Yeah, just hang on a second, Mike, because we haven't white balanced the camera. That's part of it, but just hang on a sec. Okay, I'm going to try to do a white balance on the manipulator. Yeah, just careful of that. And some spot that's as white as I can get, like that spot there. What's that? No, I was going for that part of the manip down there. I just don't know that I can get close enough to it. Right there. Yeah, right there. Okay, that's as good as I can do. Okay, so let's see. Once. Hey, Chris, is there some reason why this white balance thing won't work for me? Oh, it's doing it. Okay. Okay, now it's a little better, I think. It's a little green, but it is green. What can you do, right? Okay, that's as best as we're going to do. Okay, so okay, we just attempted a white balance as best we could. I mean, it's showing pretty true colors. That red tape is red, the blue tape is blue. the terrain is greenish because we're looking at it through water, and that can always be color corrected. But it is what it is, and it's basically, if you were to do a true color correction on that, it would be much more rusty looking, much more reddish looking. So it's clearly losing some of the red in the water column. Okay, so Chris, let's go take a look at that site that's rusty over there to the right. Yeah, yes, please. Okay, Rose, you need to just stand back over there, please. Okay, because otherwise there are going to be too many of us in here. So please. Yeah, just stop there for a sec. Okay, that's in focus. Okay, so Rose, what do you think about this spot for slurping? Okay. Okay. Okay, let's keep motoring around it. Can you go right or left? It doesn't matter. Pilot's choice. But we want to sort of circumnavigate it a bit. Okay, I'm not so worried now about the headings and whatnot. A, things have changed probably in 15 months, and B, we just want to try to find. I understand what you're saying, because in the picture that Craig sent, I could clearly see the sort of knobby botrytal sort of matte material with the iron on it, right? So that was what I saw in the picture. So let's just go around it a little bit to see what we can see. It looks a little less active to me than it did in December of 19. Is okay. Yeah, that's good. So just keep going around. So the pilot is we're now heading 109. It's 109, and we're just sort of lateraling around to the west a little bit and to the north. Yeah, so just keep coming around. This one, yeah, that one right there. Okay, so let me just try to focus in on that. Can you get my arm out of the way now? So that it's looking at something useful rather than blocking the whole field of view. Okay, all right. Okay, step back for a sec. Thanks. All right, so, and yeah, over, keep going, keep going, keep going. Okay, and up, please. And I think that's probably good enough for now. Okay. Oh, there you go. Okay. Okay, so let me just zoom in on this for a sec. So here I'm just zooming the camera in to try to get a close up and focus. And the controls on this camera are, hold on a second. Yeah, right. Okay, so there's the bacterial mat. The whitish stuff is the bacterial mat. Now, Rose, does he want the bacterial mat that has the iron on it, or the iron sort of staining on it, or the bacterial mat that does not? All of it, of course, right? Spoken like a true scientist. I want all of it. Okay, so yes. So that whole thing, we want to just get that in the slurp chamber. So basically, so get the slurp hose out. So basically, Jason has a vacuum cleaner of sorts called the slurp. Yes, that's right. So do whatever you have to do. We found this site. Now, before you do it, can you actually get the temperature probe out and let's measure temperature? Because you can see right there that there is shimmering water that is coming in right in the center of the screen. So that's where the flow, sorry for all of the motion of the camera, I'm trying to line it up here. Okay, so you see all that shimmering water. So that water is clearly a little warmer than the about 2 degrees centigrade ambient bottom water here at 2,500 meters in the East Pacific Rise. And that's what we're trying to sample for Craig Moyer at Western Washington University. Can you guys take a 4K? Okay, you are. Okay, don't take a long one, because if he gets 30 minutes of this stuff, then he's seen it all. That's fine. Okay, 30 seconds of it, that's fine. Okay. No, it's all right. Hold on a second. Okay, that temperature probe that Chris is pulling out, we just had a watch change here. So we've got a new watch team here. Oh, just to relieve, oh, that's right, okay. So that wand-shaped device is a temperature probe that it allows us to measure temperatures both at low temperature vent sites and high temperature vent sites. Now, Chris Judge, who's the pilot now, is going to be nestling the ROV front of the basket into that outcrop. Hold on a second. Right there, right there. Pretty sure, let me go and zoom in on it again. Yeah, you see down on my screen? See, it's all coming out of that crack right there. Got it? Okay. Mike, is that now clear? Now that we're doing close up? Mike or CL?
Yeah, it's pretty clear. Right now, excellent.
Great, okay. Thank you, CL.
It's pretty amazing, I have to say, blown away.
That's my wife, CL, who has put up with me doing oceanography for over 50 years. Okay, so we're going to be measuring temperature. You're going to be seeing it on the Alvin T probe. So let's read it out so that I can also let people know what we're measuring. And he's going to try to put it right in that diffuse flow there. I'll zoom out a bit to give you some context of the outcrop that we're working at. It's hard to say whether that substructure is, it looks to me like it's lava, but it may also be just jumbled sulfide blocks that are that basically have sort of tumbled over after the as the chimney grew. So we're now up to 4.8, 5.1, 5.5, 6, 6.4, 6.7, 7. Chris has the probe tip right in the place where the shimmering water is coming out. 9.2, 9.37, 10, 10.5, 11. Billy's here. Is this what you're looking for? Is this too low? This is too low for you? Okay. We have Billy Dowd, just relieved one of the other Lehigh students. Billy is also from Lehigh. And we have Charlie Holmes from Texas A&M University, Jason Sylvan student, who was out with us the last couple of times as well. He's doing his PhD work on some of the hydrothermal relic time hydrothermal features here at the East Pacific Rise. Okay, so we're up to 15 degrees, 15.2. Okay, I think we're good on temperature. So you guys note that about 15.3, 15.4 degrees C coming out of the shimmering water right near where we're going to take the sample. Okay, slurp hose, please. Yes. And we only have one chamber, so we're just going into the regular slurp chamber, single chamber. I honestly don't know. Do you know what filter they put on it? I don't know that it does. I think it just has a flapper valve, and we're sucking into that chamber. What's that? Well, the volume is about, it must be about 3 liters in that chamber, right? And do we have a camera that focuses on it? Do not? Okay, so we're just going to have to suck for a little while. And does it suck fast or slow? Do you have control over the suction? Okay, so before you start sucking it, let's make sure that it's not. Yeah, I would say that that's probably not a bad idea for these things. The bacterial mat is not going to escape on us. So she wants. Here. You don't need any samples here, right? Okay. Okay, Chris. So you see my dot here. So get that guy first. Oh, yes, it's that. Yeah. See, it's that. So start with that, and I would say position the hose over it before you start to suck, and then we'll see how we do and start it low, right? Well, we'll see what it takes to get this stuff off the rock surface. So hold on a sec before you start sucking. Does that look in focus, everybody? Yeah. Okay. So that white stuff that you're seeing is bacterial mat growing here on the surface. Okay. So let's get a little closer, and then we'll give Ben the word. So we've got Ben Pietro on the controls again here on the right seat. Okay. Yeah. So I would say go ahead and suck and see how we do. Okay. And flush it? Okay. What time is it? Okay, I still got time. And where's Sentry? Over there, right? Yeah, fine. It's happy, fine. Okay, and up a little bit. That's good. Okay, so I think you can give Ben a word whenever. Yep. Okay, so I think it's got to need more suction. Yeah. Yeah, we need definitely more suction. Are you seeing current going to the motor? Put it a little closer. It's barely, it's not really doing anything. I would turn it on full power if this is what we got for suction. We don't have any suction. Okay, now we have suction. So let's hoover all of that stuff up to all of the whites, all the white base stuff. Okay. No, just keep going. Akel loves vacuuming. So we're getting all of the white stuff, Akel. Yeah. If you just go on my screen and look at that. Okay. Okay. Let's go and get it. We want all of this stuff. Yep, there you go. And then down all this stuff here, and then we can move up to get that stuff. So there you're seeing the slurp sample vacuuming up the bacterial mat. That's good. And then, yeah, you can drag it along this stuff too. All of that material is going into the hose and then into a chamber that's around a gallon or so in volume. Yeah, especially the white stuff, Akel. The whitest yellowish stuff. There's, yeah, the stuff right here. That stuff right there. There you go. And right here. And there's, can you think you can reach up there to the right? Okay. this stuff here especially. Oh, is that right? Okay. How about there in the left-hand corner of the screen? Down low, yeah. This whole stuff right here. There you go. And down here, and then we'll call it quits. I think that's plenty. And then down here. Down here. Yeah, just vacuum that surface right there. Okay, and then keep going up there. Maybe a little down here. Okay, good job cleaning up a Raquel. All right. Now, what do we have to do to make sure this doesn't come out? It's got a flapper valve on it. So basically, we're done once we start sucking, okay? Okay, we're done sucking, done slurp sampling. Slurp sampling noted and logged. Okay. Okay, we're finished here. We're going to next go up to Q vent. So that little target right there. It's about 100 meters or so just up to the northwest of us. It's a vent that has been right up on the axial trough wall, the eastern axial trough wall, and was active for many, many years. And then after the eruption became inactive, but then recently it's been reactivated as the volcanic cycle has sort of progressed since the last eruption in 2005, 2006. Did you take any of the 4K of that? Yeah, good. Oh, we have a marker to put down. Okay, so after you stow the hose, you got it. Okay, there's a marker in the bio box. You'll note that there's lots of bungee cord and yellow loops. That's what the pilot uses to be able to secure things on the basket so they don't flop around and get in the way. And all of our samplers are strapped down the same way. So it's a very creative, low-tech approach to making sure that things are secure on the basket so they don't complicate the operations. And the pilot has a variety of cameras that's helping him to be able to see things on the vehicle, because remember, we're not looking directly at the seafloor. We're looking at the same flat screen that you are back home. It's in there. It's in the black bio box. So he's got a bunch of different angles to sort of simulate, if you will, peripheral vision and the way in which the human eye is understanding sort of spatial relationships and depth of field when you're looking at a 2D screen. Oh, sorry. And there you see the box opening. That'll give you a pretty good view of the temperature loggers that are in there. There's a marker in there, a Raquel, that says Zeta Garden. And they're rubber banded, so you can try to stretch them out somehow, catch it on something and stretch out the rubber band. That's the starboard manipulator coming into view. The ROV has 2 manipulators. There you go. And there's the Zeta Garden marker. These are made out of syntactic foam and then a short piece of both polypro and some cable, some steel cable that's attached to a few galvanized shackles. Again, low tech, but pretty effective. And if you can try to put it behind some rock someplace, but anywhere right up here, right around where you were sampling is great, Kel. I wouldn't even worry about shaking out the loop. Just leave it there. That is perfect. Okay, there you go, Zeta Garden. Yep, hold on. No, close it, please. We will eventually, but not right now. We need to have some a shackle or something on that thing so it doesn't. It does, except when it opens faster than what you can grip the yellow loop on. All right, good. Get the arm out of the way and I'll take a couple of pictures. Do you take a short 4K of this? Yeah, right now. Tell me when you're on. Okay. Okay, I'm taking a couple of the stills. Okay, we're done. We're out of here. Let's go. We're going to Q vent right there. Q vent is pretty much directly north of us.
We're just gonna go north 60 meters. Good.
Yes, yeah, we're ready to go. And can you fly just sort of up and over this, Raquel, as you're going to head north pretty much?
Now, I talked to Raquel and he says that every time elevators come up, it's been coming up in different degrees.
Is that the Setri team? Let me talk to him.
Yeah.
Here.
Here he is.
Hey, is this Zach or Justin? Hey, Zach. Okay, so now we've got another sort of white chimney structure. We don't have Sam here, right? But let's go see if we see Alvanella in here, and let's go see what the temperature is of that feature. That may be a separate vent. You're trying to focus on the, oh yeah. So okay, so it's got worms. It looks like it has alvanella. It's got 2 worms on it. Let's see if we can measure a temperature there. This is just a little bit north of Zeta Garden by maybe 10 meters or so. So zoom out so we get a view of the whole thing. And turn down the ISO a little bit. Okay, so actually frame it there, take a picture. It looks like it's got pretty good flow coming out the top. And I would say probably as long as we're here, we should try to sample a chunk of it if we can to see if we can get some worms for Sam. This is it's a no-name vent. It's about 7-10 meters north or maybe even less of where we just slurped at Zeta Garden. So it is a chimney. It's venting. Let's go find what the temperature is and see. We want to maybe look around the other side of the orifice for a second, or the other side of the chimney for a second, and measure the temperature. There's clearly a lot of sort of smoke diffusing out of the top of it. I would say, what do you think, Akel? Maybe a couple meters high? Yeah. And it has that nice little beehive with some flanges on it. So this is definitely a higher temperature venting.
Hello, Dan. This is Jill.
Hey, Jill.
Hi. This looks a lot like the vent that we slurped alvanellids off on our first dive.
Yes. Yeah. It's got that same sort of characteristic, and it's got what seemed to me to be some juvenile riftia, but definitely alvanella on it. Well, which I think Sam would like. We're going to try to go around to this side. I know it's going to get smoky here in a minute, but we want to sort of circumnavigate it and see if we can see any particular vent structure. And then what we might do is just take a sample of the top and see if we can then make an orifice that we can measure the temperature in. Does that sound okay?
Yeah, that sounds great. Thank you.
Okay. Yeah, thanks. There's this, so Akel, this knob here. Can we try to go a little closer to that and get that in the field of view? Yeah, it's going to be tough, Zoe, because it's shimmering so much to focus on it. You can try to see if you can do a focus on there, but it's going to be hard. I mean, that's not bad. That's good. That's better, I think. Yeah. Just go ahead and zoom out. And there's the sort of black structure around there. So this has multiple flanges on it, lots of alvanella, and these decorated little chimlets on top with flanges and little beehive structures. So I think we want to see, Akel, if we can find some place where we can measure temperature. I mean, I think if we broke off the top of this, we'd probably get some flow, right? Well, that's my guess. Although I'm wondering, I mean, there's this little black beehive right here. I'm wondering, what's that? There is stuff coming out of it. I mean, there's definitely stuff coming out of it right there, right? But it's definitely diffusing. But all of those little red things, can you turn down the ISO a bit? That's all alvanellas in there. Turn that down. Is this a place where you can set up right here? Because we've got worms here. If you could grab a piece of that rock and put it in a bio box, which manipulator are you going to use, you think? Okay, let's do that. Let's put the temperature probe out and see what's going on here. Hold on a second. Hold on.
It's on shutter right now.
I know. Yeah, I'm sure it is. Can you do it in stick lock and just hover here and grab this? I mean, so where's that coming from? Where's that black smoke coming from? That's coming from underneath the flange. Okay, what's that? Yeah, well, hold on a sec. So let's put the probe right there, okay, where it's coming off the side here. Okay. So we're trying to get set up here to take a temperature. These worms that are coming out, you might be able to, if you zoom in on the worm, right, Zoe, zoom in on the worm up there. Yeah. So go all the way in on the worm and try to focus. At least gives you, okay, so now try, I know it's shimmering, but try to see if you can't get the worm better in focus. Down that, this worm, okay. Oh, he's camera shy. That looks better, I think. Yeah. Okay, so come out. Oh, he's out again now. Okay, that's good. No, you should be stopping that. Okay, that's fine. Take a still. What time is it?
It's
5:30.
yeah, 5:30.
Okay. So we've got that pretty vigorous flow, Jill, down at the bottom of the screen that we're going to try to measure the temperature of. There's that beehive that's sort of just above it and to the right in the image that you're seeing. Anyway, we'll see what the temperature is. Open up the ISO a little bit.
That sounds good, Dan. I'm most interested in this temperature here because it's smoking.
Yeah. Okay. So we're at 11 right now, but he's not close to it yet. Okay, he's just moving the tip into the flow right now, and we're at let's see. You're gonna have to go down on it a little bit, Akel. So it's at 15, 31, 49, 65, 70, 71, 69. So hold on a second. He's still getting set up on it. 642, 39. Getting colder, getting warmer, 69, 86, 99, 108, 114, 116, 108, 93. Sam, you're back here? Yeah, good, okay. But this may be the temperature that is in the range that Billy's interested in. But it is coming out from underneath that ledge there. So it's pretty tough in terms of trying to figure that out. 121, 124, 131, 133. Akel basically has a nozzle sort of up against that little ledge, the little shelf there. 153, 154, 158, 162, 168, 167. 167, 169, 167, 168. Okay, I mean, is that good enough, Jill, in terms of characterizing the temperature here?
Yeah, I heard 169. That's the highest.
I think 169, was that right? 170. Well, you got up to 170. So yeah, of that order, right?
Yeah.
Okay, all right. So I think we're good with a temperature measurement right here. Note that please in the log and in the files there. Do you have 4K of that? Okay, hold on. Just hold it there, Kel. You can take a grab. Yes, you did. Okay. Okay. All right. Now the question, Kel, is zoom out a little. Here, let me just have that for a second. Sorry. We'd like to try to get a piece. Sam, you want to look? There's alvanella all through this, so I was thinking we've tried to get a piece in the bio box. Is that all right? I mean, we may get chemistry here later, but might as well get some worms. Yeah. Okay. So there's all of these worms here. The question is how to best give you a chance to get a bunch of worms in a rock, right? And I don't know if it's like grabbing this ledge. Can we go and look more carefully at that? So Sam, what do you think, please? Okay, so Sam, look at all the holes and look at all the worms right there. Right? So there's at least 1, 2, 3, 4, 5 worms there that I know of. And probably more. If you could get, you think you could grab that whole piece? Okay, so I would say let's try to see if we can grab that. Yeah, okay. And whichever swing arm you want to put it in is fine. Mike or CL, if you're watching, or anybody, or Jill, is that view of that too bright? We're trying to gauge what it is for us versus what it is for you in terms of contrast and exposure.
It looks a little bright.
How about now?
That looks better.
And you can still see the detail because we think it's in focus.
Yeah.
Okay.
Yeah, it looks good. Looks good, Dan, I think.
Okay, all right. We're still getting used to dealing with this 4K camera box and adjusting, shutter iris and the ISO and all of those combinations. Are you still recording 4K? Okay, for Sam, for the sampling site. Okay. Okay, so we're getting set up to try to take some a sample of this flange here of the sort of triangular piece that's sticking out towards the ROV for some alvinella worms for Sam Vosen, one of the PhD students in the Santiago Herrera's lab at Lehigh. You think you can come in from, yeah. Do you have the buy box open? Okay. And the pilot is looking at a bunch of different cameras to give him a perspective of how far in he is underneath that little ledge and how we might grab it. You got a piece. Oh, there was a big fat worm. Yeah, let's hope they all stay in there. We can go back and get a couple more. Well, you made another smoker anyway. Good job, Akil. Thanks. That's good. Okay, let's go back and get another piece, please. Which Sam, where do you think? I mean, there's alvenellates in this guy up here, right below where you have the fingers in my screen. This piece right here, Kel. This one. Let's try that one. I mean, it's got a bunch of tubes on it. I see one alvenella in there for sure. And there's a displaced one underneath it. And there's this guy who obviously wants to come home with us. Hi, yeah. Hey, Zach, what's up? I'd like to have a half an hour. We'll try to do what we can with it. I would prefer, I mean, it's 5:45 now, right? Okay, so I mean, I was hoping that I'd be able to continue working while you're starting to come up. No, you have to. Okay. Okay, that completes our sampling here for Sam. We're going to, oh, look, wait, is that one gonna fall in? Is it? It is. All right, very good. There's one worm that was swimming around and it made the mistake of swimming right into the bio box. Okay, all right. So we're done sampling the worms here at this yet unnamed site that's just about 7 meters north of Zeta Gardens. And let's, yep, seal it up, please. That's the whole surface of this chimney right here. Am I muted? I'm muted, right? I'm not muted?
No, we can hear you, Dan.
Okay, good. All right, so that we just panned up and that's the surface of this sort of bulbous structure that's a couple of meters high, where there's a lot of alvanella worms. I'm assuming all of those tubes are alvanellids and not tevnia. Any biologists want to weigh in on that? They look an awful lot like tevnia, but I don't know those sort of. What's that? Yeah, you think they're tevnia? I mean, they look like it. They're certainly not riftia here.
You're talking
but
about
there's an awful lot of the few.
You're talking about down
low.
Dan,
Yes, down low. Those little tubular, structures that are coming out.
Yeah, I did not think so.
Okay, you think they're all alvanella tubes?
I think they're most likely.
Yeah. Okay. All right. Okay. So at this point, we want to try to. Where's Sentry on our map, please? To the west. Okay. All right. Okay. Can we just go north to Q vent, please? I have 10 minutes, so let's go north to Q vent and see if we can get there. Yeah. We're trying to coordinate the recovery of Sentry, which is coming off the bottom here in about 15 minutes. And that's what we'll be doing next. So for about the next hour and a half, we'll be just waiting in position about 50 meters off the bottom so that we can have the ship recover the autonomous vehicle Sentry. So this is the chaotic collapsed terrain in the floor of the axial trough. So you want to go directly north from your position. Yeah. Okay. No, I'm talking to Akel. Sorry. And a lot of that sort of brownish staining that you're seeing on the lava surface is all sort of hydrothermal clays and debris, if you will, that's fallen out from the venting in this area. Okay, what's that spire we have in front of us? See, this is where I was getting confused when we were here on our other dive, and not really understanding this structure, which is about 50 meters south of the position of Q vent, but in fact it may be. Jill, you there? Am I muted or not? No. Okay. So
you're loud.
Okay. So we're now, at least based on our map, in the historic position of Q vent, about maybe 40 meters or so south of it. But clearly there's some vent chimneys here, and let's see if it's smoking at the top of Kel. It looks like we have a smoker at the bottom right here, right? Is that smoking down there? We're 20 meters south of Q vent. Okay, so this may in fact be Q. So can you guys please note that we're about 20 meters south of the historic Q vent position, but this may in fact be Q vent. It's got lots of chimneys on it. It's got some activity, and it has this other one that's just a little bit to the north, up a little bit further on the wall. A little too bright. Dial it down. Okay, that's good. So it does have active smokers. Oh, actually, could we go up there, like Akel, right there? See what's smoking on that little chimney, that orifice right there. Okay, let's zoom in on that, Zoe, and let's look, because it looks to me like we're seeing the same sort of whitish, but not quite as much as we did at Bio 9 and P. But you know, it looks like it may be phase separating, so it may be hot. It's almost clear fluid coming out of that chimney. I don't know if Jill, if you're there, if you can see that. So in this case, I think you need to fiddle around with the iris a bit, and stop it down a bit. So that looks like a pretty productive. That one right there looks like it's pretty productive. You think you could get the temperature probe out and stick it in there? We got maybe 10 minutes. Sorry, I mean, you know, I'm doing the best I can to coordinate all these vehicles. Sam, where's Sam? He left. No, I'm just, his friends are here, you know. Got more Alvanella than you can shake a stick at around here. So we're just stopping here for a minute. This may be Q vent. We're going to try to stick the temperature probe in, and then we're going to have to break off and go. That's looking pretty good, Zoe, in terms of focus and stuff. Can you zoom in on those worms and try to see if you can focus on them? Yeah, so it's a little off focus. So just, yeah, stop it there. Try to see if you can get them to pose for you. Almost had it. That's good. Stop. Stop. Okay, zoom out. Stop. Okay, just give people a view that. Stop, stop, stop. That's good. So that's the shimmering coming out of the top of this chimney, and the albanella worms with those reddish, multi-fingered worms. So what do you call the fingers on an albanellid? What is it called? Tim? What are those wavy tentacles?
You can call them tentacles.
You can call them tentacles. Okay, go zoom in on those guys. So do you want to tell people what those things are doing, Tim?
Yeah, those are the tentacles that surround the buccal orifice or the mouth of the worm. So it extends those out there, and I believe it's scraping or wafting over microbes that it will eventually eat, and they're also sensory. So they're sort of like feelers, if you will.
Great. Okay, Kel. So we'll try to get a temperature measurement here. You connect me to the Sentry van. I want you to take a temperature. Let's see if we can get the temperature probe in that flow, and then we'll yes. Just, yeah, right, that guy, the black orifice there. Just see if you can put the probe in there to see what kind of temperatures we're getting out of this guy. Because if it's super hot, then this may be another one that we'll want to do.
Hello, Dan. This is Jill.
Jill, you have to hang on for a second. I'm talking to the Sentry van, sorry.
Okay, no problem.
Sorry, Jill, go ahead. Take snaps. This is great.
Yeah, I was
You
going to
taking
ask.
some video, some 4K video? Go ahead. Yeah, go ahead.
Oh, I was going to ask, how far are we from Q approximately?
I think we're there. We're about 20 meters away from the position, the historical position, and that's certainly within the slop, as far as I'm concerned, of the positioning from LBL nav with Alvin and whatnot. So I think this is probably Q, and so clearly it's become reactivated, and it's got some pretty good flow up at the top here. I'm assuming you can see that in the video. We're trying to take a temperature measurement of that little chimney that's on the left side of the screen. So he's going to try to zoom in on that a little bit. It's hard. There's a lot of particulate matter. There's just a ton of sort of clear fluid flow out of here. It's so we're going to try and get a temperature measurement of this stuff right here. And Akel's doing a good job of making an orifice for us. So we're up to 61, 73, 83, 92, 100, 105, 108. But yeah, no, that's okay. So it's a very small orifice that he's working with here. So what's that? It's right there in my screen. Yeah, right there. It's probably not much bigger than the diameter of the temperature probe tip. $51, $72, $92, $111, $127, $142, $154, $164, $172, $180, $186, $193, $205, $212, $219, $225, $234, $244, $250. I'm measuring temperature. I'm reading out temperatures here. $268, $271 in degrees Celsius. $279, $282, $283, $283, $283, $284, $284, $285. Do I hear $286? $280, $285, $285, $285. 285 is the highest we got. So it's of that order. I think we have to break off now so I can go recover Sentry, but I think we got a pretty good reading that it's 285. Might be a little hotter someplace. We've only been here on the hold on, let me get oriented. Right now we are heading 34, so we're here on the south west side of the structure. That's the image of the very top of the structure, and the structure is probably a good maybe 4 meters high. Akel, what do you think? 4 or 5 meters high from the bottom of it? Okay, and it's built up right against the wall of the eastern axial trough. And so we pan over to the right, please, and over to the right you see another. There's white stuff there, so clearly there must be some chimneys there. It's a little hard to say. It doesn't look like it's that smoky, but there's some bacterial mats and whatnot, and alvanilla probably. So it does look like it's smoky. Good job, Zoe. That's great. So there is clearly some activity there as well, Jill. Can you see that?
Yeah, I can.
Okay. All right. So we got to break off. We got to go collect our AUV, and it'll be about an hour and a half until we get back on the bottom. And the next thing that we're going to do is we're going to go get the elevator that we positioned here, and then we're going to go do the detailed sampling at M. So, and there's tube worms there. Very good. So, way to go. All right. So there's clearly some activity right there, and we'll probably have to come back on another dive to do this. But okay. So, Jill, you have any questions for me at the moment?
Nope. I think we should go meet Sentry.
Oh, okay. Tim, thanks a lot. Everybody will be back in about, as I said, about an hour and a half when we've got Sentry safely on deck, and then we continue with this dive. We'll be diving until about 6:30 tomorrow morning, local time. We're on mountain time here, and then we have to recover so that we get it on deck at 8 o'clock tomorrow morning during a watch change for the Jason group, and then the biologists are going to put all their gear on and they're going to take a dive tomorrow. All right, I'll be back shortly. I'm going to go get something to eat. Okay, did I? I'm good now? Yeah, okay. All right. This is Dan again, and just as a little update, we just got Sentry back on board, and we're going to now get back the control of the DP on the ship and then head to the bottom. We are going to be going to the elevator site, which the elevator landed pretty much where we guessed it would, maybe 50 meters or so further east, but it is at the latitude of Q vent where we left some equipment on the bottom. So we're going to go to the elevator, grab it, and then bring it back over to Q vent to get the tide gauge, and then go up to M vent. So we're going to go down to the bottom and make the traverse, so we can see the bottom and again do some photo traversing. Because we've got some sampling bottles on the elevator that we're going to switch out eventually, right? And we have when we go to do M, we're going to need to take a gas tight in our hand and maybe stick it in the black bio box or something, because it has no holster because we didn't want to confuse things further on the basket for you. So I have 2 gas tights on the elevator and 2 double majors. And the majors are going to get swapped, and those are taken. Yeah. And just so you have some advance notice, it'll probably be on your watch. So when we have taken these majors at M vent, which is where we're going first to take fluids, we're going to go back to the elevator, put them in upside down to some extent. The snorkels, we rigged up a special basket for each of them, and the snorkels are going to go down, or the springs are going to go down, and the snorkels are going to hang over the basket. And we rigged the straps so that they'll fit. We checked it all out, and we're doing that because there's so much gas in the fluids that we think that the bottles are coming up sort of half full, and so we're hoping that maybe that'll allow more of the fluid to stay in and not escape. That's Jill came up with the idea, and we figured we'd give it a try. So if we do get more fluid that way, then we'll maybe want to do that again. But I can't do both things. I can't have baskets with the upside down mounting and the regular mounting, because otherwise you wouldn't be able to get a hold of them. So okay, we're going to the elevator right now, and I'd like to traverse at about 5 meters with the down looking camera on, so you could turn that on. And are you guys all set with the watch change? We're getting there? Okay, great. And Zoe, watch your back as you go out with your backpack, please. Here, Sam. Go ahead. Oh boy. Yeah, thank you. Yeah, well, who's with me? Keldon's with me, and Charlie? Okay, yeah. So just remember the tide gauge. Sort of like remember the Alamo kind of thing. We're going to carry it with us. I'm not sure where it's going at the moment. we're trying not to abandon it. Oh no, are you serious? You don't have a spare pair? No, but yeah, I know, but your regular reading glasses, you don't have a spare pair? You don't have a spare pair of your reading glasses? Go get, oh. Well, you sure? Okay, because we still have time before we're doing anything. If you want to go look again, seriously, we got plenty of time. Okay, all right. Yeah. What's our altitude? That's me. Sorry. What's that? Sorry. What was the altitude? Okay, great. So we're going to go down to 5, Mario, and then if you could put my camera on. Thank you. Thank you. Okay, so you note that in the log, Kelton. The down-looking camera's on. And the port manip, if you get a chance. Oh, sorry, go ahead. If you could just swing it to the left a little bit. Yes, yeah, so we want to push that out if it's in. And then over to the right a little bit. And a little more to the right. Yeah, that's great. Good. Yeah, but with the starboard manip, could you come just you can come up a little bit and to the right a little bit. I'm just trying to, yes, sorry. Yeah, I'm just trying to get rid of the shadow that I'm seeing on my manip clam. I think that's okay. Yeah, let's leave it at that for now. Thanks. Yep, all set. And I'm fine going as fast as you're willing to go. I know it's not that far, but okay. And I think this is you, right? So I think you're all the way over to the left. So square yourself up. And whenever you get a chance, Mario, if you could slew the starboard arm over so it's not in our field of view. Thank you. That's great. Perfect. Thank you. We're getting the elevator. We're going back to Q vent. Going to grab the tide gauge and take it. Thank you. And take it with us up to M. We're going to take the whole elevator tide gauge pair up to M on the east side of the axial trough wall, position it there, and then deal with sampling M. Oh man. And we could come down another meter, Mario, whenever you get a chance. Just try to keep it around 5. I think that'll make it a bit more visible for folks that are watching on the Zoom channel, as well as for us to see what's going on. So we're right now just going over the axial trough. These are lava pillars and collapse ramparts as part of the 2005-2006 lava flow. Very sort of chaotic volcanic terrain that's has these beautiful archways between the lava pillars. They can be anywhere from a few meters high to up to 5 to 7 meters high. And all of that jumbled lava crust that you see is all the surface of the flow that's all collapsed now, just under its own weight into the trough floor. That's not bad, Sam. That's a pretty good altitude, Tito or Mario. Thank you. You're what? Yeah, you're 8 meters right on your port side where the altimeter is, but that stuff in front of you is pretty close. Yeah, because you're just coming over you're just coming over the trough wall. That matches up beautifully with the bathymetry data. And from now on, it should be pretty much low baits. I can't remember, but I don't think we're going over any lava channels. Yep, that's fine. Is that your alarm, Tito, or somebody else? Oh, good. Oh, you mean black screen? Great. What's our heading? 053. Okay, so we're heading up to the northeast to get the elevator. We've got 100, maybe 180 meters to go. So we're now on the low bait terrain on the east side of the axial trough. This area that we're going over, if you could see the bathymetry map that was made by Sentry over the last couple of years, is an area where the axial trough actually is has a little bit of a transition, and it's transitions from a very sort of well-defined trough that's about maybe 40 meters wide to an area that is a bunch of fissures and collapse areas. We think this is one area certainly that was impacted by the last eruption, and it transitions into a narrow sort of trough to the north, up where Zeta Garden is, and that continues up to M vent and to Biovent. North of Biovent, the axial trough really tapers out and has done so. And since we initially mapped this with the Argo system with Rachel Heyman and from UC Santa Barbara in 1989. Yeah, probably. Can't you guys just write it in bigger letters or something? Tell you what, I'll be right up. You guys are going to the elevator anyway. I'll be right back. Okay, we've got about 15 meters to go to get to the elevator. Mario, the pilot, just turned and is heading for it. And we've got Tito Colassius, who's the expedition leader in the van as the navigator, and Gabby Inglis, who is the engineer, sitting to the right of the pilot. And in the science watch, we've got Sam Vosen, a student at Lehigh University, Keldon Peer, postdoc at Lehigh University, and Charlie Holmes at Texas A&M University. So the elevator that we're heading for, this device that we use to ferry samples up to the surface and equipment down to the surface, down to the seafloor, should be coming into view shortly. Has a big yellow block of syntactic foam on the top, which provides the flotation, and it's weighted down by a big block of steel plates that can either be mechanically released or released by an acoustic signal that's sent from the control van here in the ship. If you put my arm, the port arm up, I bet you'll be able to see it before we see it with anybody else. I think it's right there, straight ahead of you. I can just see it on the top screen of my camera. See it right there? Yeah, there it is, right. Did I leave my the pointer thing? Oh, great, thank you. All right, and we don't have to do anything to this. All we have to do is just grab it and then carry it over a couple 100 meters over to Q vent, just outside of Q vent, and find a nice spot for it on the low baits. We're pretty high above it right now, because the way that Jason carries the elevator is they put it in the manipulator and they put it in front of the basket. There you can just see the top of it coming into the field of view on the camera. And then they traverse over the seafloor at about 7-8 meters so that we just keep it in front of us. And they do that because otherwise it would be a possibility of it getting entangled in the cable. So we like to have it below us. But it's easily maneuvered and manipulated by the starboard manipulator. Okay. So the deal is that they control the heading, but you can control the thrusters.
Yeah, so I'm controlling X.
Right. And the azimuth, right? Yeah. Okay. Yeah. That's what Akel was showing me earlier. Right. Yeah. Yeah. Right, and I don't know if it matters, and I don't know where the wind is coming from. But from Q, we're going to go to M and then violent. So all of our movement is going to be north, just to keep that in mind in case it helps. Yeah. So You're going to see the starboard manipulator now come into the field of view, and the gripper, and he's going for that bail, that ring on the top of the foam block. Those devices there are various antennas, a radio antenna for when it's on the surface, a flasher, that red device with a ring around it that's in the foreground is a USBL beacon, and then it's got an iridium beacon, a satellite telephone beacon, the tall one on the radiator hose on the right side of the block. Okay, so now we've got it, and he's going to come up, and then we're going to head west. So we're getting set up to sample M-vent. That's what we're ultimately going to be doing for probably about 3 hours here this evening. It's one of the primary events that we're interested in and that we followed for almost 3 decades, since the early 90s, since we've been studying this part of the mid-ocean ridge, this fast spreading section of the mid-ocean ridge here in the East Pacific Rise. Did Okay, so we were able to just switch the feed for a second. It's a little distorted just because of the zoom sort of format. But what you're looking at is the high resolution 1 meter bathymetry map made by Sentry, the AUV that we have on board over the last couple of years of surveying that we've been doing here. Ross Parnell-Turner and Jun Nai Wu. Jun Nai is Ross's PhD student, have been working hard on that along with Greg Curris at Seafloor Investigations, a longtime collaborator of mine. So this bathymetry has proved to be really exceptional and very well co-registered to our previous observations and event sites. And that's one of the things that we're working on as part of this grant. Having this very high resolution bathymetry of the volcanic terrain before the next eruption will be crucial in us being able to make some really quantitative assessments of how much lava came out, where it went, how thick it is, and all of that. So because knowing the volume of a seafloor volcanic eruption is very useful in terms of understanding the sort of volcanic processes and history, if you will, that is going on here at this fast burning ridge. But here what you're seeing is the two red circles are basically the window that we're using in this ROV single body mode, where we're affectionately calling it the donut. We're flying at the perimeter, if you will, of the donut. The purple rectangle is a beacon that is some distance up on the fiber optic cable that's telling us where the end of the cable is. We float the cable with these syntactic foam footballs so that it stays out of the way and is up above the vehicle. And then the two yellow dots are the ROV and the elevator that we're moving over to the west. And then you see over on the west-hand side of the map, there's this linear green, blue, deep cleft. It's actually about 8 or so meters deep. It's a center point and about 40 or so meters wide. That's the axial summit trough. That's this narrow cleft at the plate boundary between the Pacific plate on the west and the Cocos plate on the right. And that's where the majority of the volcanic and hydrothermal activity is occurring along this section of the ridge. So it just gives you an idea of how we're navigating things and where we're going in general, and how sort of well controlled, if you will, our targets are for when we're making observations and sampling. Okay, Charlie, that's great. Can you switch it back now to the seafloor view? And the grid lines on that map are about 100 meters apart. So we've got about another 180 meters or so to go. And we're flying up a little bit above what is the observational distance, at least on the forward camera, because that's blocked by the elevator. But my down-looking camera, which is flashing every 5 seconds, is taking pictures of the seafloor as we're moving along here to the west and getting high-resolution 24 megapixel still images of the volcanic terrain. we're making a straight line track to the west. We've got another 100 and maybe 20 meters to go. We're going to be picking up an instrument that we left by QVent, a tide gauge that we put out here. We deployed it in December of 2019. And we're going to be recovering that hopefully soon, perhaps on this dive, but it's basically trying to collect all of the various gear that we have here and bring it back so that we can analyze the data. So we're after that, we're going to head north, not very far, another 50, 80 meters to MVent, and that's where we'll be working to take fluid samples and install temperature loggers to measure the hydrothermal fluid temperature over the next course of the next year. We have another cruise, the second out of 3 cruises that we may be doing with Alvin in about a year from now, next March. We're basically doing this in a sort of a staged way to understand how the temperatures and the chemistry of the vents are changing over time in a period where we predict that maybe just close to the next eruption. In general, the eruptions, at least historically, we've observed that there were 2 of them, one in 1991, 92, and the last one was in 2005 and 2006. And so we're at the point where it's likely that there might be another eruption based on the spreading rate here at this fast spreading ridge. It's spreading at about 11 centimeters a year. So we're using this as an opportunity. We just happen to be sort of at the right timing to be able to look at this in detail and use some new ideas and new instrumentation to investigate these linked magmatic, volcanic, and hydrothermal processes. Yeah. It's Okay, so we're about 30 meters or so from Q vent and where we think we left the tide gauge. We're going to go collect it and bring it back to the elevator and then head north to M. Those open milk crates are the ones that have the 2x4s on the right side there. You see that?
We're going to empty the one.
Yeah, it won't fit in that one. The feet need a rectangular one. I got out the pictures that I took of it when I made it and mounted it on the Alvin elevator, and so I have a better recollection now. We're higher up than I thought. Kabash. Okay, it's there. Perfect. Okay. Now, if you could get down closer to the seafloor, Mario, and we'll go look for the tide gauge, which should be pretty much just, about 30 meters or so west of where you are now. Do you need to drop a target here? Or?
The elevator.
Okay. All right. Okay. Sorry. Yeah, so note that in the log, right? Yeah, it's right on the 78, right on the 78,800 latitude XY. And tell you what, can you turn off my camera, the down camera? And why don't you pan around to open up the iris a little bit? So we're now looking for the tide gauge, and yeah, open up the iris a little more. That's good. Open up more. There you go. Good. That's good. Yeah. I know the video isn't great. We're about 4 or 5 meters above the bottom, and we're continuing to go west to find where we left the instrument. Could you maybe put the port manip up a little bit so we have a little bit more of a view out further afield? Yeah, that's good. And down a little bit, just down a little bit more. That's good. Yeah, that's good. Okay, great. Yeah, yeah, these are all big collapses here. I mean, we did leave it on a place that was, you know, a good solid low bait, but I remember that there was a collapse near it. But you know that there's lots of collapses around. What's the left? What's the left? Right there, right in front of us.
Pretty sure it's on the side, that real shape.
Right there, right forward of you. Yeah, just go drive straight in my camera. What's that guy? Nope, that's a vent chimney. Okay, that's a vent chimney right there. Yeah. Okay. So we don't want to go there. We want to. You're going to want to stop going forward. And, yeah, that's Q vent right there. I think. Yeah, I think he actually has to come. Yes. Right. Yeah, because we were there and we went exactly where you're spinning now, off to the left. So you want to come left. It's someplace on this side. Yeah. Yeah, I don't think we decided to. So I would, let's see, you're right now headed south. Okay. So why don't you just go a little bit further south and then go here. Let me show you on the. I wouldn't go much. I wouldn't go much past that dot, and then turn left. Right there. That right there. Okay, and now I think go ahead and turn left. Okay. What's in front of us? That's the elevator right there. Okay, that's fine. Yeah.
They logged that it was north of Cuba.
What's that? Is it a little north of Q vent? Okay, well let's go back to the elevator and go a little bit north of Q, and then like maybe go like
where we saw Cuba and then
yeah. Okay. Okay, we just double checked, and it turns out that we left the tide gauge a little bit further north. So we're going to go grab the elevator again and then position it up at MVent. $78,891. 78,891 is right there, and 4394. 4394 would put it right there. Almost looks like it was right on this little ledge right here. A little north of the cursor. Yeah. Yeah. And basically we'd want to put the elevator just a little bit east, 10 meters east of that cursor. Yeah. Oh, sorry. Yeah. Okay. And then go to goal to cursor. Yeah, right. Yeah. Okay. And to stop it. And just dial this down. Yeah. Well, right. Right. Yeah. Okay. So we're just going back to pick up the elevator, and we're moving about 100 meters to the north. Thanks, Mario. I know this. I hate these mice. So speed it up now again, that he's got it. Right. Yeah, right. Yeah, okay. And to get a range and bearing, I go to coordinates. To get a range and bearing, I go to coordinates. No? No, no. Oh. No, but I guess what I was saying is when you guys draw a line to figure out where you're going and what the range and bearing is. So what is that? Map? Oh, range tool, that's what it is. Yeah. Oh, so from to there, click here. I hate these mice. Yeah, right. Okay, so 97 meters at 335. You want to sit back down here. There. Is it falling out of the claw? Is it not keeping the drip? Yeah, right. Right. We're having to adjust some of the hydraulic settings for the manipulator so that it locks in on the bail on the elevator. Okay, we've got the hydraulics sorted out here, and we're now traveling up to the northwest to put the elevator down by where we think the tide gauge is, just near M vent. We'll pick up one of the Lily sampling bottles at M vent it's on the elevator now, and bring it over to M vent with us, so we have the full complement of samplers we need for sampling that vent. Yeah, I'm pretty sure it is. Okay, we are at M vent. Could you just turn the music down just a tiny bit? I'm not sure, but
I noticed that same thing here.
yeah, right. So it looks like there's a bunch of collapse right there, and you're heading north, right? More or less. Yeah. Okay. Yeah. So I would say you definitely want to move a little bit to the right. You want to move a little bit to the right. That's going to be your best chance of low bait terrain. That's that the elevator isn't going to drop down into the trough. We definitely don't want to go left. We want to go right a little bit. Can you open your ISO? I just want it over to the right a little bit. You see the tide gauge. See, that's definitely the tide gauge, and it's right near a collapse right here. And so we need to maybe put the elevator someplace over here where it's a little, there isn't a big collapse, but we just need to check below us. Yeah. Because it's a little hard. It's a little hard to say right here. I mean, it looks pretty solid. Can we get no, that's right. I think we'll be able to see okay with my camera. I mean, that's looking pretty good. I'm just worried about what's off to the right. Off to the left, this is all okay. So let's just get a little closer. I mean, can you just get closer so you're not dropping it from high up? Okay, great. So let's try right in there. I mean, that looks solid to me. It doesn't look collapsed at all here. I just don't know what's here. Yeah. Yeah. I mean, in other words, if you just went in that neck of the woods right here and kept going down, I think you'd be in pretty good shape. Although, no, that's collapse right there, Mario. See that here I'm putting my pointer right there. That's collapse. No, you got to go right. Yeah, this is definitely the wall of the trough. Yeah. That's what I thought. We just put it over the edge, the tide gauge. So and this whole area here. So stock work. Jill reminded me stock work. We got to look for stock work, and that's down in this nasty part of the inner east wall that we're going to go look for it. Okay. But yeah, you definitely want to go right by 4 or 5 meters, Mario. Yeah. And I wouldn't come up too much because there isn't anything over there. There's no big things you're going to run into. It's all going to drop off to the right, but very, very gradually. It's very flat right up here. And you're going to keep wanting to come right. We're going to need to get away from this collapse right near the wall. Yeah, you can just lateral right like that. That's beautiful. Perfect. That's the tide gauge. Yeah, I would keep going. No, we don't have to go faster, but we just definitely need to keep going right. All right. Yeah. Okay. Can we stop here for a second and go down? I think And what about this place right up here? This looks good, Mario, right up here. Stick it on this piece of low bait right here would be perfect. So that's a little bit forward. Yeah, we definitely have to go up there because this looks like it drops off here. So you definitely have to go forward some. Oh, it's more collapsed, but it may not be collapsed too much. But definitely up, yeah, up on, got to get past this collapse feature and go over to that part where it's more solid. Okay, so yeah, it's all. I think that there's enough solid terrain here, Mario. If you can land it right there. Is it down? Looks down. Yep. Okay. Looks good. Great job, Mario. Yeah. Yeah. So now, and the gas tights are right here in front of you, right on this side right here that's facing us. All right. So we placed the elevator just a little bit to the east of the tide gauge, and that's just a little bit east of the M vent structure. And now we're going to go and grab a Lilly gas tight sampler from the elevator, and then we'll go to M and get to work. Do you need to let Jill know, or is she not going to be up for this? I know her.
Hello, Dan. I'm here.
Ah, you're there. You're not snoozing.
I'm not snoozing. I'm here.
You're not snoozing. Okay.
Don't
yeah.
we share?
So we so we had a little bit of a sort of not wild goose chase, but I had forgotten that I'd brought the tide gauge up to M. We thought it was a Q. The watch standards did a great job of looking back in the notes, and so didn't lose too much time. But we're so now we're well positioned. We're within a few tens of meters of M vent. We've got the elevator here and the tide gauge here. We're just going to go down and pick up one of the Lilly samplers so that we are fully loaded for taking the fluid samples at M.
Okay.
It needs to be focused.
Great.
It's not in focus.
Thanks, Dan.
Yep. That looks better, Sam. Yeah. And it doesn't matter which one you pick. It's those guys right over there, one or the other. And they're in scabbards, so if you can pull them straight up and out, that would be great. Right, and the bungee goes in that sort of narrow slot between the handle and the bottle, so it's got about 4 inches of space that you have to pull it up. Sorry, say again. Oh, sorry. Take a picture of the basket, please. Still. Oh, you did? Okay, thank you. Maybe, do you want to open the bio box first, or you're just going to carry it in the grip? I think eventually, you know what? Eventually, you're probably going to have to do something else with it. So probably, is it okay to keep it in the gripper? Because we don't want to drop it. No, no, because we're set up. I'd rather stay set up. If we're in a good setup, I'd rather not do it. So I'd rather have it with us. Yes, yeah. It's got to go on the starboard one anyway, right? But as long as it stays in there. But I was just thinking if he opened the bio box and put it over that in case it dropped. Yeah, okay. Yeah, well, that's all right. You zoom in on it so we can see what's going on with the bungee. Yeah, it comes all the way up and over that the end of the T handle. That's it. You got it. Great. Sorry, Alvin. When is the ship supposed to come out of the box? Everything I've heard is that All right, we've got the Lily gas tight, and the vent is off to your left side. So that's where we're headed. Okay. Zoom out. Mhm. You did him. You want me to hold it, Tito? You got it? Okay. You want me to hold the light while you lift it out?
Yeah.
I got this. Yeah, there's a slot in the gray tube yeah, there you go. Yeah, so rotate clockwise. Yeah, there you go, like that. No, wait, stop. I think you got the nozzle in the plastic tube. So you got it. Yeah, no, yeah, right. So you got to come out, right, and then that's the slot, and that big long metal piece fits in that slot. There you go. There you go. Yeah, like that. Right. Yeah, that's got it. There you go. Great.
So I guess you gotta power that box.
You want me to get down there, Tito? Sorry. This is going to the port. And here's the new one. Got enough cable here. If you want to hand me the other one, I can. Okay, got it. That's that one. Got it. Should I put this one back in the box? Jill, if you're still there, we've now sorted out the manipulator issues. They had to put in a new control box and recalibrate it. So we're should be lifting off and heading over to M vent, which is just a few tens of meters away to our northwest.
All right, Dan. Glad to hear that got fixed.
Yeah. So let's go to M, and we're going to need to look around a little bit and figure out what orifices we want to target. So we don't want to go, once we get there, I don't want to go very fast. I want to be able to look around because we want to try to identify the structures that we have traditionally sampled, as well as the one where the temperature logger has been left, which is, I think, on a little sort of shoulder, just a little bit north of M, like, 7 meters or so, or 10 meters north of M.
Sounds good.
Sorry, Sam. You all right? Oh. And if maybe Mario, you can move my camera sort of forward and up a little bit. I think that gives us a pretty good view and helps us with how we. Yeah, and then over to the left a little bit. Yeah, that's probably pretty good. Thanks. You want me to set that up for you so you can see it? That good? Like that? Okay, I think you might want to come a little bit more left. So that we're looking into the trough. There it is right there. There you go. Now we're talking. Excellent. Okay, Mario. So let's go take a look at that, please. All right. So the tide gauge is sitting there just to the just on the rim of the axial trough wall, and there's one of the M vent chimneys right there. So Sam, you focus and get the white balance sorted out on that camera, and you see there's these other ones that are off to the right. But let's have a look at this one first, and let's see where it's. You okay? Shoot. I'm sorry. I apologize. Right, so let's just do a slow pan up, Sam, to see if there's activity. Yeah, you could shoot some. I mean, I know we have the gas tight right in the field of view here, but we're doing that for safety reasons. What's that? Yes, it was, but they forgot to put the target in, so I had to zoom in on the manipulator, blast it with light, so it's a partial white balance, if you will. And I believe that this, Jill, is the original M vent structure.
Seems very tall. I mean,
yeah.
I always thought it was short.
And I guess I'm also saying that because the place where we had the logger installed was down by the new flows that are just down to the basin to the left of it, and that's where the logger was, and the lava flows just barely came up to the top of it. So let's continue to look around, but I don't think that this one is active, but what's active is just a little bit to the a little bit to the north. Right now we're heading sort of west northwest,
Okay.
and as we go further into the AST, there are some other chimneys, but I believe that this one does not have much activity. Although when we were here in 2018, it had some bacterial mat, and there was some stuff going on, and you can see down below. So let's go, if you will, Mario, around to the left, please. What's that? Yeah, over the tide gauge and over to the left. So let's sneak around and see the other side. Yes, and we've got Stefan Sievert here from Woods Hole Oceanographic. Yeah, Stefan. The old and the other one, what you were calling flea, is the one that's that way, right inside the. Little wispy stuff. Yeah. Okay, so now we have a good view. Just hold that. Take a snap, please. You recording? Charlie, good. Okay. All right. So now you're seeing a sense of the structure built right on the eastern wall of the axial trough. Sam, can you pan up any more to get to the top of that chimney? Maybe almost. And it doesn't look like there's any shimmering coming out of there right now. Yeah.
Nope.
But yeah, it doesn't look like it. It's certainly got some bacterial fuzz on it. And then focus it, Sam. It's got bacterial mat on it. That's good, Sam. That's great. So look, that's gorgeous.
Nice.
That is. Take a snap of that.
That is weird looking.
Yeah. Don't name a new vent, Stefan. I know, I'm teasing. But that's a gorgeous sort of, and I don't know what kind of bacterial mat that is, but it's got this gorgeous green color in it. But clearly this is one large structure, and I think that this may be the older one, but we've got stuff in front of us. Okay, Sam, now zoom out so we can get a view of a view of the vent structures that are on this little. It looks like a sort of a series of lava pillar structures that maybe the vents were built on, because that's what I remembered, is that there are some selvages, lava selvages around the sides of the vent structures. So there in the distance, we're seeing more activity clearly. And Sam, with the sample that we took here of M-vent, the big one, right? So that was one of the places where, in fact, wasn't that where the logger was recovered from? So, and you can see smoke in the distance. So let's keep going past there, Mario. Because I think what we may be wanting to look for is down, around and to the right. So just keep going around the structure here.
What's our heading,
can see the smoke.
On which direction is the AST?
Yeah, yes, we're heading west right now. So we're heading into the axial trough.
Yeah.
Okay. So we're heading west into the axial trough, and this is all this complex series of collapse features and lobate, sort of collapse structures right along the margin of the AST wall. And now you can see where the vents are. It's just inside on this sort of inner terrace that's just west of the east wall.
Yeah.
And there you can see a couple. There's Sam zooming in now on some of the smokers that are there. And we're going around to the south side. We're going around the south side of the structure that we're coming up on. That's great, Mario. Yeah, so just keep sidling around to around this left side of it. But there's clearly all sorts of activity going on within the, the sort of rampart between the vent structure and an old sort of joint between two big lava pillars. I mean, it's all right here along the wall. And that's
Yeah.
certainly what I remember of M, that it's built up on, perhaps on top of a lava pillar that's, right along the margin of the wall.
Mm-hmm.
And you can see, Jill, in the distance there, it's, it's not sheer, but there's a pretty steep slope down towards it. So, I think that's clearly the place to explore for stock work down there.
Yeah, totally.
Right. I'll take another snap of that, Sam. That's a great shot there.
This is a really neat view.
It is. Yeah, it is. I mean, and I don't think I've ever had this good a perspective of it. Okay, so now these features, if you look at that thing that's right in the middle of the screen and you look at those selvages, that's what we looked at, and where in fact some of the colonization panels were put in the ECS cruise in 2018.
Mhm.
And the logger, I think, was, the vent logger was maybe down. I mean, that's the surface of the 2005, 2006 flow below us here.
Mm-hmm.
Because those things were, and if we did a better job at, if we get closer, you'll see that they're sort of orange stained, but it's just like the series of shelf edges that go up, and that's the lava flow. That's the drainback structures on the lava flow that went right up against the vent.
Oh,
And
I see.
they're in the
Okay.
upper right.
So the
See what I'm
vent
saying?
here to the there's sort of these, you see the drain back feature, and then the vents kind
Exactly.
of shutting off on the right here.
Exactly right. Exactly.
So it was lapping up
Right.
around the bottom.
Up around the bottom. That's right. That's
Yeah.
right. Okay, but you see, as if you go, so now we're turned and we're heading 47 degrees. So we're heading northeast now. So now we're looking towards the tide gauge, which is up over, just over the edge of the AST wall, the east AST wall, and this whole complex along this sort of ridge, this inner ridge, just inside the AST, is where the venting is happening, right along the margin, which I'm assuming is because you've got a, open it up a little bit more. I'm assuming you've got, a dike below here, or a fissure that's partly associated with the sort of collapse structure and why the trough is forming right here.
Mhm.
But you see that other structure in the distance that's sort of pinning this one that we're on, and the next one over there, and the original one, right, is and then you've got this whole series of venting going on in between.
Yeah.
All along that rampart.
Yeah. Is it really
Okay.
milky here?
Oh, it's quite smoky, and there's lots of particulates in the air. I mean, some of that may be stuff that we stirred up, but in general, the AST has been, I think, a pretty particulate laden, if you will, certainly more so than in many parts of it. And you can see it's just hot here. It's still, the ambient is around 2 degrees C, but it's definitely hot here.
Yeah.
Okay, so what's smoking here, Jill, is there's a chimlet around the side of the structure in the sort of center and around to the left side of that structure. So Mario, can we sort of get in there? Yes. You don't think we can fit right in there? Right, but the vent to the left is the one that is the tall, big one that doesn't have any activity. We're going to want to get stuff from here. So I'd like to get a little closer and to see. Yeah, right. So, and now we're heading 115. So we're just coming up onto a southeast heading. So now we're looking down southeast along, looking almost to the south, down along the margin of the axial trough. So can you do a little better job focusing and white balancing that. Or not white balancing, but with the iris control and the ISO. So you can see the the whole sort of surface of it is shimmering.
Yep. Wow, it's hot.
Yeah, exactly. Okay, so and then go up. And the smoker is right there,
Yeah.
right? And it's pretty clear fluid coming out of there.
It's at the very top. Can you, it's a little harder to see on the Zoom feed. Well, maybe it's just because it's all shimmering, but is it that darker kind of vertically oriented smoker on the far left near the
far
top?
left? Yes.
Yeah.
So Sam's going to zoom back in now. So zoom in, Sam, and where my pointer is, center that up. Right there. Okay, so stop. Go down, pan down, please. Stop. Okay, so that's where. Okay, you got that now, Jill?
Yeah.
So it's that finger-like. I don't know how big that is, but that finger-like sort of vent that is right on the side here. That's where the main effluent seems to be coming from. So I guess I'd like to stick the temperature probe in there and see what we get. Yeah. So let's try to set up here. Zoom out, Sam, a little bit. And do you remember, Jill, you've got your notes in front of you, the temperature here at M when we were here last time in December of 19 for those samples?
Hold on a sec, I'll pull it up.
Yeah. Yeah, you see that's where I think we ended up getting, no, it wasn't there. No, you see that's a structure that's right by the tide gauge. So that's the one that's extinct, that's not active. So yeah, sure. No, I understand, yeah. So I would try to do a better job of the iris, closing the iris down a bit on that. Close the iris down a bit on that. Yeah. What are you doing? You're doing the zoom. You're not doing the iris. You were doing the wrong knob. Yeah, that's it. Because you'll get more depth of field if you stop the iris down. Okay. All right. Now zoom in just to make sure you're in focus. Zoom in. Yeah, your focus is a little soft. So focus it and then zoom out. That's it. Now out. Keep going out. That's good. Okay, stop. No, go in so you don't get so much of the basket. Keep going. Stop. Okay. Probably need to go a little bit out more so you get the top of the structure. Go a little bit out, zoom out so you get the top of the structure. Stop. Good. Okay. So snap that. Are you taking video? Okay, good. So you take one of this right now, and I would stop the lens down, and your ISO is too high. So go down with the ISO. You're on iris still. Yeah, you got to. I know it's hard because you got three things in one knob. So okay, now you see what you're doing. That's great. Just keep going. Okay, so now we want the temperature probe and we want temperature at that guy.
Yeah, and the temperature in 2019 was 300 Celsius.
Yes. Okay, hold on. So you can land down there. You can land. I mean, what's that? Anything that's not white, but this structure is pretty strong, so I wouldn't worry about that, right? Well, I mean, these little guys, you're gonna if you try to sample them, you have to be super gentle because they're tiny and they're probably pretty fragile. But down here, that part of it is solid. Okay, zoom out a little further, please. Okay, we're just going to land here and then see how we do. So we think that structure is about 2 meters high, more or less. About as high as the vehicle. Not much. I mean, it's right there, right? Yeah, 1.5 meters or so tall. At least this active portion of the chimney above this basal portion here. Yes, please. Sam, pan up. Okay, we're getting ready to take a temperature measurement with the Jason temperature probe. Yeah, anywhere in that. Yeah, zoom in on it, Sam, to help. In that clear water right at the orifice, if you can. And that piece, when you touch it, might break, but you know, that's the way it goes. All right, we're about to go in. So we're up to 5, 6, 7, 8, 11, 12, 13. He's still hovering above it, but he's just trying to line himself up. 19, 20. Yeah, that's okay. 64, 65. 45. Yeah, that's looking a little better. It's a pretty small orifice, Jill. It's venting pretty vigorously that with the clear fluid, but we're at 31. Can you put the nozzle in there?
Go ahead and touch it. If it breaks, it's okay.
Yeah, no, exactly. If you could stick the nozzle, we're at 69. I would go ahead and put the nozzle in there, Mario. Yeah, just jam it in there. 71, 75, 78, 82. Yeah, that's okay. Yeah, we're making a sampleable orifice. That's fine. Okay. Yeah, I would. Yeah, right. So just push it back away from you. Yeah, right in there. That's it. Back a little further. Yeah, right there. Okay, jam it down in there. Okay, that's good. So 115, 121, 128, 135, 142, 158, 159, 156, 153, 158, 164. That's good placement, Mario. 176, 182, 191, 195, 198, 198, 198. 201, 203, 204, 206, 213, 215. So one of the things that I'm thinking, Jill, is it might be worthwhile for us to just sample this guy and and then try to use the basal portion as the orifice and see what we get. Because I'm guessing that it's I'm guessing that we're losing some through that little chimlet.
What do you mean by losing some?
I think we're losing some heat. I don't think we're seeing the max temperature up in this portion. Just even though it might be, I don't know, a foot tall or something like that, I think probably it'd be better to get rid of that top part of the chimney and just sample it, toss it in the bio box.
Yeah,
let's just work.
Excavate that black chimlet.
Yeah. Right. Okay. So let's pull the probe out for a second.
Can.
Yes. So we're going to want you to stow that. Oh, there you go.
Oh, there we go. That works.
Okay. Zoom out, Max. Let's see what we got. Okay. So now we have a more manageable structure. Yeah. However, I think what you could do, but before you go and sample that, here's my preference, Mario. I'd like you to stow the probe, sample that, put it in the bio box. Right. Put it in that. Put it in the forward box, bio box.
Oh, you want me to grab the chimney?
Yes. Yeah. With the left-hand manip if you can, and then stow it in the bio box. In the bio box in the basket, right, in the coffin. It'll even if we put another sample in there, it's identifiable enough. But it's interesting the character of that fluid coming out of there.
Yeah.
You see how it's streaming, Jill?
It is. Yeah.
I mean, it's either got a ton of gas in it or something.
It seems, yeah, it looks really unusual.
It's very unusual because it's super clear.
It's super clear, but it's also really hot.
Yeah, zoom in on it, please. So you see that, I mean, it almost looks like it's not, I know it's not flaming, but it's clearly coming in sort of waves, and the shimmering, it's picking up the shimmering of the backdrop, if you will, but
yeah.
Focus. So I want you, well, you could just keep holding the gas side as far as I'm concerned, because you're going to do everything with a port manip, right? You're going to grab it and put it in the, yes. Can you not open the bio box with the port manip, or you can? Trying to focus through the shimmering on the just the right side. Yeah. Zoom out. This is M vent, yes. Well done. Excellent. Okay. And yeah. Okay, so let's use the arm to grab that chimney down at the base and just, just jiggle it. It's likely to be pretty friable, but yeah. Because what we're hoping to do is create a nice solid orifice right at the base of that chimney where, okay, now zoom in on Sam. And it looks like this knob is in front of you and that chimney is behind. So if you could grab it right here at the, you see where my red dot is? Right. Yeah, down, like down here. Grab it as far down as you can. And I believe that that chimney is behind this knob. That's sort of this softball sized knob. That there's a little bit of space between there and there. Yeah. Can you open the grippers anymore? Yeah, I would go in a little wider. There you go. That's it. Yeah, no, you're in front of it. What's that? Yeah, exactly right. This one right here. That's right. That's what you're going for. Yeah. That's it. That's the perfect one. And a gentle close. Oops. You have any of it or not? Oh, you had some of it. Is that it right down there? Well, I think from my point of view, it's probably better, Mario, to focus on. Let's stick the probe temp and try to get a better temperature in there now. So let's get the T probe again. So we now have something that's a little bit closer to the base of the main part of the structure, and hopefully we'll be able to get a more accurate temperature.
Yeah, in your view, can you still see where it's venting?
Yes, absolutely. So Sam, zoom in on where the venting orifice is.
Oh, I see it. Yep.
He's got it pretty much centered in. There's so much shimmering going on. I know it's hard to see, and we're having a tough time focusing.
Yeah, I see it though. It's where the really vigorous shimmering is.
Yeah.
Yep.
It's nearly impossible to focus with all the shimmering going on. Sorry.
That's okay. Looks like an impressionist painting here.
Yeah, right. I mean, I think I'm pretty close to being in focus.
Yeah. Yeah, that looks better. You can see some
Yep, hold on.
alvinellids and scale worms.
Yep. Hold on a second. So you see I think it's this top one that's right at the bottom of the screen where the chimney was. I think that's the one you want to go for, right? Yeah, exactly. The one you're heading for right now.
What's the temperature right there, Dan, before he gets in the flow?
7.6, 9, 35, 47. Yeah, it's right where you just sort of nicked the orifice right there. 146, 106. $70 now. And it looks like you're really just off to the left of it. It's really close to where you are right now. So now it's $38, $48, $53, $67, $59, $70, $80, $90, $100, $110, $119, $126, $134, $142. So you sort of right up next to that. Yeah. So $185, $189, $193, $196, $200, $210, $215, $216, $220, $221, $228. So it's in focus, so don't touch the focus there. $235, $236. So Jill, do you see that alvinella sort of a center left? That's coming out of the out of its whatever, or maybe it's just left there. There's another vent that is just below it and to the right. That's venting, right? So right now we're at 239, reasonably stable, I would say, and I guess we could try to go into that other orifice right by the worm and see what we get there. It's pretty steady at 239, almost 240.
Okay.
Okay, so Mario, let's take it out, and I want to go to the right here by the just to the right of the worm. Yeah, that one, right, exactly. Okay, he's close to close to going in here. Yeah, over the right, 114, so 14, 15, 21, 28, 53, 61, 70, 80, 96, 105, 124, 133, 141, 155, 160, 167, 172, 193, 200, 204, 215, 217, 220, 226, 230, 233. What did we get up to before when we measured the other one? 239. Okay, we're at, okay, so it's 240, 241, 242, 243, 244. You think you can go in any deeper on it or not? You just push it down. Push it down in just a tiny bit. It's 244 right now. And he's trying to jam the probe tip in a little bit more. No change. In fact, it's gotten a little less. It's down to 237 now. 231, 239. Yeah, I mean, it. You know, I think my guess, Jill, is that it's diffusing so much through the whole structure that we're definitely seeing, you know, some mixing going on within the chimney itself, and the, you know, the sort of primary fluid is probably hotter, but it's a little hard to tell.
Yeah. Yep. That's all right. We can use magnesium.
Yeah. Okay. So so right now we're back up to 180, 200, 203, 205. Oh, now so 271, 272, 273, 274, 270, 269. $255. What happens if you just move it that way? That way, away from us, just a tiny bit. Oh, okay, that's what happens, right? Okay. All right, now where is it? Well, now it's right. It's, yeah, it's. I think it's, yeah, I think it's still. Yeah, 152, 59, 64, 194, 198. $225, 230. We're around 235. It's not getting too much hotter, but he's moving it around a little bit. It's still around 230, 235, 236, 238, 240, 242. What did we get up to 270 last time?
274.
Yeah, okay. 249. All right. So I guess I feel like at this point we could go ahead and look around a little bit more and see what else might be perspective. We know we've got this, although I'm a little worried about how exactly we would sample this and not clog nozzles, but we certainly can try. Okay, Mario, let's come out of it. And can you fly with a temp probe, or you want to just stow it? So, okay. All right, let's continue to go around the structure a little bit to the left. Yes. What's that? It's not. No, it's not. That's right. Yeah, because that one, right? Because that one has that. Okay, and right, and that's what we tore off it, right? The top part of it. So what's the XY of that? For what? Yeah. Can you zoom way in on this guy? Kelton, read me those numbers again. Okay, wait, hold on. Okay, and then what else? 7, 8, 9, 4 point. So that's north. Okay, so here we go. So let's so Mario, let's keep going around to the left of that structure, and we want to go. What's that? Okay, well then back up and let's go around that one and then go up to north, because I think we want to go just, yeah, exactly, Tito, right. So we want to go a little bit further north. There's another structure here a little bit further north. It does not seem so. It's this white base here, and it didn't seem to me to be doing that. What's that? Oh, good. Okay. No, it seemed the structure is built up on the sort of lava, the collapsed lava on the east wall. But we're now going to go about maybe 10 meters or so, or maybe 20 meters up to the northwest. And that's where the logger had been placed. You okay with oh, do you want to do that? You want to like this? I mean, I think we're doing okay. But I mean, do you think that do you think that putting the gas tight in there would help in terms of your manipulative sort of flexibility? Yeah. No, I don't think if he lays it down that way, in other words, so it's sticking out of the box to the left. No, I think the nozzle will be okay. So I guess what I'm saying is, I think if you put the gas tight in here, I just think that would then give you a little bit more flexibility in terms of how we deal with the setup. And yeah, you've got a nozzle right there, but you can just stick that in, and then and I would say just leave the bio box open for now and just put that stuff in there. Pan down, Sam. Pan down. Yeah, there you go. All right, let's just let it live there. Okay, great. Thanks, Mario. Okay, so Jill, we're going to go a little bit further up to the northwest. It's sort of following this line of chimneys along the east wall.
Sounds good, Dan.
Okay.
That's close to the X and Y's that I gave us.
Right. So you're flying to that spot right there. Yeah. Okay. So just note that we're moving away from this structure and heading up to where we actually have the position of M vent based on my analysis of the 2019 Alvin data, which is where the logger was. So I think that's the chimney that the logger was at. But there's clearly venting going on throughout this area of 20, 30 meters right along the east wall. And Rachel, if you're there, we've got Stefan Sievert, who you were out with on one of his cruises, right? In 2004. Yeah, but wasn't Rachel on one of your cruises? Oh, before. Oh, it was way before that. Okay. Okay, so right now we're flying at 345. And yeah, out a little bit so we can see better and maybe come down a little bit. Because we do want to see, because there's something white in front of us. And so I'd like to know what that is before we go by it. So we want to see exactly how, right, exactly. Okay, so there's another chimney coming up into view. Yes, yeah, it's along the track. So let's go there and see if it's smoking. Can you do something with the ISO on that? Okay, so now you've got it lined up right in front of you. Can you come down just a little bit? See, there's that one, and then there's another one just past it. There's two of them right here. Pan to the right, because that camera's looking that way. Yeah, okay, down. I think we're still a little high. Yeah, there's two of them right off to our forward and down. You just got it barely right here. Yeah. So there's that one, which is definitely smoking. The one that we're coming up on is definitely smoking. And then I think the one past it is where we got the sample for Sam yesterday. That now we're closer to that XY. What was the temperature that we measured at the site where we got your alpha nullet piece and the chimney sample? Oh, can we go and look and see log? So there's lots of smoke coming out the far side of this one, Mario, right? So now we've got that in the field of view. And so that's good for going down. I think that's fine. Yeah. So let's circle around the other side of it. It's got a bunch of anemones on it. I'm pretty sure that that one in the background is the one we got your samples from, because we broke the big piece off. What was the temperature? Oh, sorry. All right, so we're just we're lateraling around. Right now we're heading 340, so we're almost at a heading parallel to the trough axis, which is about 350. And again, just as a guide, we've got, you know, about 8 meters below us. So these are tall structures. They're at least 8, if not probably 10 or more meters tall. The venting on this one seems to be coming from the north side. Taking some snaps too as you're going along. Charlie, are you taking any of the 4K? Okay. I wouldn't leave it on forever, but certainly, you see on the other side of this, it looks like there's a bunch of Albanel tubes and quite a bit of diffusing going on through the top of the chimney. Taking some snaps of that. Okay. All right. And then Jill, if it's okay, I think I'd like to go to the other structure just to verify that that's the one that we ripped the top off of, where we got the sample for on our last dive.
Yeah, were we able to see where this fluid is issuing from here?
I think it's diffusing out all of the top, sort of like the other one. It's just it's clear fluid that's coming out of the top with a couple little chimlets on the top.
Okay.
So Mario, let's go left still, and then let's go to the next chimney. Over. And it's right there. Okay. So it's just again, just a little bit north, you know, maybe 5 meters north, right along the margin of the axial trough wall, right? And that top there, so that smoker right there is where we're looking for, and that's got a bunch of smoke coming out the top. That's where we took the sample on our last dive when we did some exploration up here, and we want to measure the temperature there, because that's where the logger was. So this is the vent where the logger was. Okay. All right, so let's make sure that we got that totally clear in the notes. So let's get down to that level. And the vent orifice is this big one back here, which is where we ripped the top off of. And you can see, Jill, that there's a couple little chimlets that have this sort of clearish fluid coming out of them. But then there's the sort of main orifices just in back of that. Our heading right now is about northeast, about 48 degrees. We're at 9 meters altitude, and we're at the top of this thing. Yes, but we want to take the temperature there, and it's probably good if you can set up here on this side so that the smoke doesn't come in your face, right? So because the smoke's going that way, the smoke's basically going up to the northwest. But the main orifice, I think, is this feature right here. But, you know, all of that stuff down there is solid, right? If you land on it. Yeah, the only problem with this setup, though, I think, is that you won't be able to see enough of where it's coming off the back side of this. What we did is we ripped off the part of the chimney that was up here, where the smoke's coming out. So I don't know. Maybe if you lateral around and go to that side of it, go to the right side. Yeah, let's go to the other side and see how that looks, because otherwise I'm afraid that maybe the setup's going to be harder. You guys doing okay back here? Yeah, I would keep coming around. Yeah, keep coming around to the right. So we're now hitting about 215, 209, basically coming almost to a southerly heading. Yep, I would keep coming around to the right and see these orifices here. Let's see if we can get lined up on these guys. Those are gorgeous beehives up there. So I'm just wondering, Mario, this flow right here that I'm showing in my pointer on your screen here, so this one, trying to figure out where that is. I think that's up. Trying to figure out where the heck we are here. So that's the flange. That's the beehives on top of the flange. See, it looks like it's coming out like crazy, right? Right in back of there. Jill, any suggestions? I mean, we could put it in that clear flow behind these little beehives here in terms of taking temperature. You see where that black smoke is?
I do. It looks like it's really strong flow. It's hard to see where it's coming from, but if we put a probe in there, it might dislodge.
Well, I think where it's coming from, and it's interesting because you see we've got a flange pool here. You see that flange pool up at the top?
Mm-hmm.
Sam, zoom in on that, please. Right above that chimney. You see the shimmering water coming out of there. It's caught underneath the ledge. Right. Okay. Focus on that, please. You can focus on this edge right here. The other focus. There you go. That's good. Stop. Okay. So there you go. So that's the flange pool. So now zoom out, please. And I think that flow is coming out behind one of these big beehives here. So stop. Yeah. There's like, 3 beehives, the 2 big ones, and then one right in the middle of them. I think it's coming out right behind this middle guy, Mario.
Yeah, it looks like
it. Can you stick the flow back there or the probe back there and see what you get? See the black smoke is coming out behind this guy right there. So you're going to have to just sort of poke around and see whether you can jam the tip back in this flow right here. Okay, let's see. And definitely go towards the structure. Yeah. Okay, we're at 50, we're at 60. 68, 80, 90, 100, 115, 123, 153, 169, 160, 180, 190, 205, 212, 224, 231, 237, 240, 250, 260. Did you take a snap of this? 264. 266. You think you can go any more into it and away from us just slightly? 270. No, that made it cooler. Yeah, I think the main flow is more in here, right? Yeah, there and back. Yeah, oh, you can get rid of that guy. Yeah. What's that? You know, it's hard to say. I mean, clearly it's coming out of that beehive. I don't know if you can try to jam it into where the base of where that beehive was. Yeah, these little structures here, they look sort of like a beehive. No, that's what we call them. I mean, yeah, you've got, I mean, that's the flow right there where you're going in. Yeah. Yeah, I mean, I still think that the main flow is behind this big one here and to the left of it, behind these two little guys in the middle of the screen. Yeah, behind these two and just to the left of that guy. You can. Yeah. There you go. Done. Right, is this one right here? You want to try to stick it in there? Oh yeah, now you're making a good one. Making an orifice. Okay. Yeah. See, it looks like, I mean, it looks like it's basically venting through the whole structure, Jill. You know, we're excavating a bit and we're making an orifice, but it's I mean, we've got 120, 139, 145, 150, you know, but that's really the probe just sort of sitting at the surface there where you just excavated.
Yeah.
So
see how it looks when the smoke clears up.
Yeah, let's zoom out for a sec, Sam. Let's zoom out. Okay, we're just going through a watch change here, so Keldon, who else do we have for water when Billy's going to be in? And how about, do we have to have somebody come in to help with the ICLs? Okay. Thanks a lot, Tito.
Who's on watch with the water group, Dan?
So we got there's Bill's going to be here in a little bit, but Keldon said she'd stay on for a little while, and then we have Rose here and Natalia.
Okay, thank you.
Okay. So and Chris Judge is just coming on watch, along with Nick and with Ronnie. So you can see, Jill, in the video what we've got for Orth is based on the sort of dismemberment of the little beehives that we're growing there.
Mm-hmm.
And we're heading southeast. Basically, the view that you have in the screen there is actually, let me have the box please for a second. Let me center the camera up with the axis of the vehicle. So okay, so that I'm now more or less parallel to where we're heading, and that's about 150 more or less. So I'm looking down, and that chimney in the background is more to the sort of south. I'm looking sort of southeast in that view right now. And that's the one that we looked at before, and then we drove north to this one, which is about 7 meters further north from it.
Yep. Okay.
This
so
one
that was the second one we looked at.
That's right. That was the second one we looked at. This is the third one that we're looking at.
Yeah.
Right. And they're all spaced within this 10, 20 meters along the axial trough wall, along the eastern axial trough wall.
These are going to be tricky to sample.
They are going to be tricky to sample. We could take off the top of this and basically see what we get for an orifice, but I think we'll probably find lots of little holes where all the black smoke's going to come through. So that's certainly possible, but you can see what we just did. Those beehives were growing out of some small little structures. There's hold on a second, I'll try to zoom in better on what we just excavated. So I'm betting that it's and the probe tip is right up against one of them, and we're getting 70 degrees, but that's not any sort of a throat that we can feel like we're getting sort of pristine fluids from.
Right.
There's a lot of mixing going on throughout this whole thing. I guess what we could do is I could have Chris try to move to the right and go down a little bit. I see a lot of clear water coming out that the lower part of that structure. So we could go and look down there if you want. So you want to try to do that? You want to come off here? So you've got the temperature probe still on the chimney surface. So yeah, we can try to go look down there and see what we see. I don't see any other obvious places, Jill.
No, yeah, down on the right on the bottom. Although
yes,
it
right.
might not be as vigorous, but let's have a closer look at it.
Yeah, okay. Okay, we're moving around and down a little bit to the right. We're hitting about 130 right now. See, it's diffusing all the way from that lower right base, and then that knob that's down at the base too. It's got some small little orifices that are smoking.
Yep.
But not, you know, there's not a lot. I mean, I'm guessing. You can see it looks like sort of a saguaro cactus, you know, that whole part of the structure on the right. I'm guessing that there's a main part of the plumbing that's coming out of that one.
Yeah.
But clearly that, you know, down towards the base of that main structure is where the main pipe is.
Mm-hmm.
Well, probably here where you can land, but then the whole thing is on top of you. So I don't, well, in other words, you see here, you can land there, right? But then I'd be worried that you have the whole structure on top of you and the whole thing is going to fall in the basket, which will be sort of a nightmare. So I don't, for the heck of it, why don't you keep coming around, just lateral around to the right. What's going on with the 4K? Is it being recorded or not recorded? Okay, that's fine. I just want to make sure we hadn't left it on. Are you getting some snaps of this? Okay. You could record some 1080 clips of this. Right. So we can try this one. You're seeing this one, Jill, just down towards the bottom of the structure on the right-hand side.
Yeah, that one looks promising.
Okay,
that
so
little black gimlet.
Exactly. So what can you set up down below that? And let's see what we can do about that thing.
There's actually two of them.
There's two, actually three,
Three,
sort of aligned right along that. Yeah.
There's three little chimlets.
Right.
The one on the furthest left looks really nice, actually.
Okay. The one that's just coming up onto the T handle right now.
Yep, exactly.
Yeah. Okay. So that one there, let's try to put the probe in that one first. And we've got a gas tight in the bio box, just so you know. Yes, exactly. Right. Okay, we're getting set up to take temperatures at this other side. So now we are heading due north. So we're on the south side of it. Can you guys? Okay, we've got the temperature probe out and Chris is going to get lined up to try to put it in that one. You can take a clip of that. All right, so you're going for that guy. So 26, 29, 35, 41, 48, 57, 70, 99, 113, 125, 137, 170, 180, 206, 214, 222, 243, 255, 260, 264, 271, 277, 270, 285, 88, 89, 290, 292, 286, 284. Can you stick it in just a tiny bit more? I actually think that it's like that if you can. I don't know that you can tilt it much, but yeah. Down a little bit. 264, 267, 271, 76, 70, 82, 287, 291, 295. We're 300, 302, 304, 305, 306, 307, 310, 311, 313, 315, 317, 3 coming on 19, coming on maybe 20. Yeah, 320. Okay. So Jill, you said that in December of 19 it was 300 and some.
Yeah, it says 300 exactly in the notes.
Okay. Well, this guy's 322 and still climbing a little bit, although it seems to have settled out. This definitely is, I think, a better place to sample.
Oh yeah, much better.
It's still climbing a tiny bit, but it's hovering now around 323, 324, 323. What do you think? We good here?
Yep. That looks
Okay.
good.
All right. So we got
call,
almost
320.
Sorry, say again.
Is that our last call on the max T?
I think so. It's about 324, but it's been pretty stable, and he has a tip in maybe what, an inch or so. Okay, maybe a couple of inches in, but it looks like we have a good orifice and it's very steady temperature of 323. So, what's your druthers? You want to do a gas type first?
Yeah,
one of your IGTs first.
Let's test comms first before
Okay.
we pick it up.
All right. So, Bill, you okay? Okay. So hold on. Bill's going to get set up now. Remember, tell us which is the good channel. The good channel is number. Well, we have 3 and 4. Okay, and we want to check to make sure which is next to which, right? You should have a note saying. Okay, so ICL2, and we don't want to have ICL1 on. Okay. Okay.
Dan, is Keldon still in the van?
She is not, but she's somewhere close by. Why, you want us to go get her?
Yeah, let's get her.
Okay, yeah, she wants to get Keldon up too. Is that okay? Well, why don't we have Natalia go get Keldon? You stay here and see if we can get the comms going. Thank you, Natalia. All right, and we're at 324.8 at the moment, and it's been steady. Thanks, Chris. So I'm wondering. So just be careful how you move that, because when you do move it, okay, that's fine. Go ahead, go. When it moves these cables and then it wigs out my thing here. There's too many fucking cables here. Oh, sorry. I guess I need to watch my language. Yikes. Okay. Okay. And is there any chance you can point my camera differently or not? Oh, the tempo. Damn it. Right. Okay. Foiled again. Okay. Yes, yes, absolutely. Jill, do you want me to hand the mic over to Bill, seeing as how he's controlling the computer?
Yeah, that would be good. Thanks.
Okay, hold on.
Good. Hey, Jill.
Hi, Billy.
All right, so right now with IGT3 and ICL2, it's not spitting out the gibberish, but I'm not getting a good reading as of yet.
Are you getting
It's jumping
any
up to, yeah, it's jumping up to 129,000. And there's a, I guess a default error that's like -1 #J. And Kelton's back, so I'll just hit.
Okay, thanks.
What was
Bill.
that?
I'll just
I'll just hand it
Kelvin.
back to her.
Thank you.
Okay.
Oh, okay. Hi, Jill.
Hi, Keldon.
I was muted. Okay, so we haven't picked up the IGT yet. Okay.
Keldon, right now we're going to check if the comms are working well.
Okay.
It doesn't matter which bottle you talk to, but
Yeah.
I think Billy was talking to 3.
Yeah, so right now 3 is it's getting a reading, but I've yet to see a reasonable temperature.
Do you see the battery voltage?
Battery voltage is fluctuating between 0 and 12, so there's a few good ones.
All right, that's good.
They're not corresponding to good temperatures. We're getting like 90 and then like 3,000 and then some really, really off ones. But yeah, the closest I've seen to what should be like a 2 or something, right, is 90.
Yeah, it's probably not 90. It looks warm, but I don't think it's that warm.
No.
Are you seeing #WB3 at the beginning of the line?
Yes, yes.
Okay, so it is talking.
Yeah, and the battery volts is there's a couple that are like in the saying 13.4 volts.
Okay.
So it's connecting a few times, and then some of these other readings, the battery volts is like 0 or a really large number.
Yeah. Can you try talking to number 4?
Yeah. Number 4 is giving a signal, but let's see, I'm getting, oh, I just got 1.69, 1.54, and now it's jumping up to 100. Okay, so number 4 is giving a few, I think, real temperatures scattered in with
nonsense.
Okay, Dan's asking if we're ready to pick up a bottle. I think we could start with 4.
Yeah, 4 seems like a better bet here. Let's go. Are you still seeing 1.7?
Sorry, what was that, Dan? 326. Okay, Dan says they're steady at 326. So should we go? I think we should go ahead with IGT4.
Yeah. Are you still seeing temperatures on it?
Yeah.
Okay. Yep. Let's go ahead with 4.
Did you try the other ICL before I came up here, Billy, or just this one? Just two. All right, but two's the top one, so yeah, I think we can stick with that one. Okay, yeah, so I'm getting a couple like 5 Celsius. Yeah, it's hitting about 5.6 a couple times in a row. So I think that might be. Yeah, 5.8 with 13.4 volts. So I think a few of these are real temperatures. All right, so they need to bring in the basket a little bit in order to be able to take the bungees off of the new configuration.
Roger.
All right, so they're moving IGT-4 into position. All right, I'm getting more readings of 7, 8 degrees Celsius. Yeah, I'm just starting.
Keldon, did you start recording the temperature file?
Oh no, let me make a new file really fast. Good call. One sec, Dan. Okay, so we are in the orifice. Trying to figure out which of these numbers. Let's see. I think it's jumping around a lot. I'm seeing a couple 230s.
The number we're looking for is about 324.
Okay.
Looks a little to me as though the tip of the IGT is behind the chimlet, but I think my video quality is not quite as good as yours.
I'm getting it. I got a 324 and a 307. No new readings yet. In 303. $300. I got a 300. Um, Okay, I haven't seen a temperature that seems real in a while now.
Are you still seeing the battery voltage, or is it at
Yeah,
zero
it's
now?
jumping, but I'm seeing a few that are 13.7.
Okay.
That one was just 13.7, but it was at 7 Celsius.
Look, we might just be out of the flow, actually. They might be real.
Okay, that one's at 13.7 at 110. Yeah, Jules, you want them to reposition the probe a little bit?
I think so. I think it, let's take it out again and then go back in.
Yeah, if you can reposition it, that might be worthwhile.
Could we zoom in a bit on the video so that the center of the image is more where the tip is? There we go.
Rose, can you zoom in a little bit and maybe move it down a bit? Yeah, like
Yeah.
that.
Thank you.
Yes, please. Thanks, Rose.
Thanks, Rose.
Yeah, so I'm getting temperatures that are like 112 right now, I think. Okay. Okay. They're going to reposition and see if they can't. It looks like there's a slight orifice just like maybe a few inches to the right. I'm not sure how well this is going to show up in the Sulis camera that the feed you guys have. We can see it from another angle.
Yeah. Okay.
One sec. I'm mainly reading around 10 Celsius right now. It's up to 60. $115. I got 115 just now. Yeah. No. I got 113. Okay, they're gonna try repositioning again.
Okay. Can you have Rose zoom out a little bit?
Rose, can you zoom out a little bit? Thank you.
Like that's good.
Good. $112. $173.
Okay, let's hold it right there.
$270. $309. $305. $308. $270. Well, $320.
Great. See if they can hold that position, Keldon.
Okay, can you hold that position? Would that work? All right, they're trying to.
Okay. Let's get one more good reading, and if it looks 320-ish, go ahead and sample.
Okay, Roger.
And tell them that you're going to take the sample and then press sample. Rose, can you pan down?
Right.
Oh yeah, you're well, can you pan out? Or, yeah, thanks. It's going to be hard to see that turn, but how's it going, Keldon?
Okay, I think I just got a 335.
All right. So go
Okay,
ahead.
all right, we're taking the sample.
Thank you.
All right, I have the green bar. The green bar is moving. I have positive milliamps.
Good. Are you seeing any real temperatures?
Not yet.
Okay.
They can see the motor turning. I'm not sure if it's clear on your side of the feed.
It is definitely not clear, but that's good to hear. Thanks.
She likes the view. Yeah, you okay with the view, Jill?
I'm good. Yeah.
Yeah. Mm-hmm.
Any temperatures, Keldon?
I'm holding for ones that have the the 13.7 volts. Okay, yep, motor started, so now it's closing. At 15, 14, 13, 12, 11, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right, it stopped. I lost the file near the end of there, so I'm going to abort.
Did it hit 700 million?
And then do a, it was at 0, so I'm gonna do a quick close.
Okay.
Not getting a milliamp reading. Let me try that again. Did I hit the right button? Oh, okay. Close, closing. Oh, 700. Okay, there we go. So now I'm fully closed.
Good.
Okay. Yes,
right.
good with that one.
Did you press abort?
I did, yes.
Okay. Tell him to stow the IGT.
Okay, they are stowing the IGT.
Thank you. Nice work.
Okay, I'm gonna sleep this and start a new file. Oh, I have to close the file. Okay. Close file.
Thanks, Keldon. It's hard to interpret when you have to figure out which temperatures are good.
Okay, so they're gonna do the double majors next, and then the Lily gas tight after that.
Okay, thanks. If you want to head back out of the van, that's fine.
Okay.
Thanks for your help.
No problem. We'll talk to you later.
I'll talk to you later.
I want you to just forget about it for now. Yeah, exactly. Sorry, that was my screw up. Okay, so whichever major pair you want to grab. Yeah, that would be great. Yeah. That's a good one. That looks good right there. And it may help to line you up on, because you're sampling that guy right there, right?
So concerned that we're gonna get the major in a good spot.
Yeah. And it.
So this one, it makes sense to just deal with not having a great ground view, position it so that the nozzle, when we trigger it, actually pushes this way.
Yeah.
So if we come around to the left and grab the major and sort of position it so the nozzle is here.
Mm-hmm.
Give us a better chance at
We can certainly try it. You know, whatever you think is going to be the best setup without the smoke coming right into your face, because that was, I think you were right. That is a concern, but let's see what we get. Right. Can you take a snap of that? Thanks.
Well, it's hard to tell with the comm stand, but I think that was a good sample.
Yes, I think so too. I mean, based on the fact that we were able to steady it up with a T-probe first, and then I guess Kelvin said it got up to 330 or something like that.
She said 335. Yeah.
Right. Okay. All right. So we sidled around a little bit because Chris felt like he'd have a better angle, especially because we're going to be sampling with a start with the nip here. In terms of how. Sorry, go ahead.
Now we have bigger snorkels.
Yes, exactly. Yeah. So and it's not a big orifice for sure.
No.
But we're sort of behind the ledge. But we'll see when we line up on it here. You guys okay back there? Yeah. Sorry, I'm sorry, I didn't hear. What's the matter? Yes. Okay, fine. I thought we got 324. Yeah. So you see, in this view here, Chris, I mean, there's that guy, and then there's this guy, and we just sampled that guy. And the question is whether we might be better off sampling the other one, but let's. Yeah. Is that not too far away? Yep. Sure. You can go for whichever one you want. There's the guys in the back and the guys in the front. Whichever one you want. I mean, doesn't matter to me. Whichever. Yeah. You gonna grab the back bottle first, or yeah? And you don't have to re-bungee it after you put it back in because we're so close to the elevator. I think we're in pretty good stable position. Open the iris up a little bit and pan down so we can help them see it. Down, the other down. What's that? Yeah. Okay, we've got bottles 1 and 2. Let me just borrow that for a second. Yep. Okay, you're going to pull up the ICL program. Okay. All right. You think we need to get the paddle a little closer? Or you're okay. It's pretty steady. Good. Okay. No, we're trying for the same orifice. Even though it's pretty small. I mean, we have the other one that's smoking too, but
Crazy. That's why find ourselves shooting ourselves in the foot if we just stare at the wall. So we were.
I think zoom out a little bit because I'm not sure. I think that that one. It's the one in the foreground. See, I think it's that one. No, were you in there or? Because I think you were right. I think it's in here. So I would start there for sure, because I think this is a concave surface here. So I think it's there somewhere. So 13, 9. So let's see. It looks like it's coming from right there. But it's tough for those double snorkels. So we got 50. Got 106. It's an old reading. It's not getting anything right now. Yeah. I think so, yeah. It's not a very well-behaved orifice, that's for sure.
Yeah, so you've got one of the snorkels is going to be right in the back.
Right. 47. No, it's an old reading. Did you come out of alignment? Yeah. Okay, now you're going 120, 82. $30, $40, $70, 97, 133, 197, 228, 236, 252, 264, 270, no, what was that crap? 270. Okay, 286, 288, 294, 300, 308, is that right? 308, 312, 313, 314. Okay, remember where the spot is for the next bottle, okay? And I can't give you a better view of this, Jill, because the bottle's blocking the view from the camera. So
It's all right.
312. What's that? Okay, hold on. Let me try. Let me try. But it's 312 right now. Yeah, I know. I see it, but
It's like a perfect.
trying to zoom in.
It's fine.
Yeah.
Oh, there.
Okay, so that's
yeah.
okay. You see, hold on. I'm just trying to. So that's where it's getting the temperatures, and it's 315 right now.
All right. I think that's about as good as we're going to get on this little orifice.
Yeah. 311. And it did get up to 315. I guess I'd be loath to reposition it. The only thing is I can't really tell. No, the bottles are venting because I can see that on my camera. So they are venting.
Good. Okay.
And if you, yeah, they are venting.
And what's the ICL say now?
We 310.
Okay. Yeah, let's take that.
311. Can you move my camera a little bit so that we can see the springs come up? Because I think that's going to be oh no, we can see it on yours. No, never mind. 316. Let's just hold it for a second if you don't mind, Jill.
Yeah.
Okay, it's steady at 316. 317. 318. 317. You ready to trigger, Jill?
Yeah, let's do it.
Okay, let's go ahead and trigger. Yeah. 319. It's moving a little bit. 309. The springs are. It didn't move a whole lot, but when we moved it, the probe tip is now down to 246.
Yeah. Take a note of that watchstander.
Yeah. 252. It's coming back up. 259. So did you note that in the log, Bill? That when we triggered the Tom Thumb, that the temperature did go down. Okay. It's 267 right now. Oh, what happened? What did you do? Yeah. Hold on. Okay, 274. And as far as the springs are concerned, it looks to me like they're almost up. I don't know if they're still creeping up or not. It's hard for me to judge. What do you think? I think they're done. You think they're still coming up? I think they're done. I'm wondering if does the left bottle, is it just a perspective I'm looking at it at? Probably, huh? It is still coming up, isn't it? Yeah. So it looks like the left bottle is coming up slower than the right bottle. Number one is, so make a note of this. Number one, the spring is coming up much slower than the right. So I don't know exactly what could cause that, but the ICL temperature is up to 274, Jill.
Great. Okay. And the high temperature was 319.
Bill, what was the high temperature? 319? 319.
Yeah.
Yeah, it's. I don't know. It doesn't look like it's coming past that other spring, right? Okay. All right. I think that the bottle spring is up right now. The left bottle spring is up. Yeah. Okay. So we can pull it out and let's put it back in the saddles.
Thanks, everyone.
And let's do the other one. Jill, so question for you. There's another little orifice just up from this one that looks like it might be a little better. I'm wondering if we can try to take a sample from there. It might be a little less small, a little larger diameter.
Yeah, I think that's fine. That should be the same fluid. They're so close.
I mean, it's not more than a foot away. So
yeah.
Hold on, let me zoom out here. So let me give you a view of that. So it's.
Is it that one that's kind of right in the center? Oh.
So hold on. So where we sampled, Chris, was back here, right? Was in back of this little ledge here. That's where the black is where we sampled, right? So in the view that you have right now, Jill, hold on a sec, let me make it a little better. In the view you have right now, I have in the center of the frame, okay, right in the center is where we took the sample.
Yeah.
Okay, and if I zoom out a little bit, there's another one that's up higher.
Yeah.
That's got a bunch of fluid coming out of it. Now I'm not so sure looking at it from this perspective whether it's better or not, but it is pretty vigorous, and it does look like maybe the orifice is a tiny bit bigger, but you know, I don't know if we can fit both nozzle tips there. You can see the mineralization around the around it. I mean, I would try to get one there and see what happens.
Yeah, it might be better defined right there too.
Well, that's what I'm thinking. Okay, all right. So we're going to stow this bottle and pick up the other major. But again, this one too looks like it's phase separating.
I don't know.
You don't think so?
No. We'll see.
You think it's too low temperature
Yes.
to be doing that?
Yeah.
Yeah, but well then it's got a heck of a lot of gas in it. This whole business with the clear fluids that are coming out, you know, very vigorously. It definitely is a different character than P and Bio 9 for sure, where you had those jets of white right at the base of the black smoke coming out of the orifice.
What I think we are also seeing, you see how it looks a little bit steamy almost?
Yeah.
I wonder if it's forming little particles of anhydrite as it heats the seawater.
Maybe.
And normally that gets obscured when the fluids are also making sulfide particles.
Uh-huh.
In this case, this fluid, for whatever reason, it's quite hot, but it's carrying very low metals. So it's not forming particles, it's not forming as much smoke.
Right. Yes, the second double major, please. Can I sit for a second? Ow. Oh my God. Oh Lord. Oh yeah. Just took a couple of Sula stills. And we're not taking any more video, right? Okay, good. I think it's going to be a bit of a trick to stick the temperature probe in here, but we're certainly going to try. Yes, we want to do the upper one, both for the double major and perhaps even the Lily sampler. We'll see what we get for temperatures. Okay, we've got the other double major in the claw. Chris. Uh-huh. Yeah. You have to have the secret password. Oh, come on. Okay, so don't destroy the orifice just yet, okay? All right, so you're way up on top there. So let's zoom in a little bit on the orifice. Yeah, I am too, because it's like barely the size of one nozzle. Forget two nozzles. But let's stick the probe in and see what we got, and otherwise we'll go back to the other side. There was a trash on the ICL thing. I mean, it looks aligned. I want to tap it in.
I'm gonna come up and rotate away.
Okay. Okay, just cleared. Oop, too much, too much, too much, too much.
So retract it all the way. So we can only rotate out of the way, but that's sort of the gap we want to make up there.
Stop, stop, stop. Yeah. All right, you're almost on the surface there. Nothing? Yeah, okay. All right. So now we're okay, we're just getting lined up on the Tom Thumb for the ICL. All right. You know what? I think that top one is not going to work. It's not big enough. Let's go to the bottom one. Jill, we're going to go back to the same one that we just sampled because the top one is too thin, or it's too narrow a diameter for both snorkels. I'm afraid we're going to get a compromise sample in one bottle or the other.
Okay, let's go to the original.
Yeah. So that one's pretty much in the center of your view there. Yeah, I think right back there, right, is where you were. $50. Really, you think that's it? No, I mean the left side nozzle is not even close. I think you got to go back towards the wall and right a little more. You're at 50. You can't go down any on it. 72. 101. No, 92. No, I think you need to go still back, and it's not a great orifice for sure. I know. No, 58. $115. Where did you just go? More to the right? 147. Right. So it's 143. 129. Temp probe's right between them. You can't go out a little more into the chimney. Yeah. 156. 141. 112. Oh, rats. Let's see here. No, it's not going to do it. Not sure why. We're not in the sweet spot here. What's that? Oh. Yeah, I see what you're saying. So we got to keep those hose clamps tighter, guys. Yeah, that. Try to go right there. And I was going to say, see, I think it's right there, right? It's right there. It's in towards the face of it. It's running right up against the wall that you just excavated. 100. 210. 225. 239. 237. Damn it. Come on, 201. No. 224, 223. 257. 248. 236. Maybe, yeah, maybe try that. I don't know that you need to go more to the right. 248. It's not much, yeah, so stop there. And can you go, no, we're at 189. So no, clearly we've got to be down right in it to get so we're not entraining the seawater. Yeah, try that. Yeah, try that. $160. 173. Right there. I know, but it's like the diameter of one of the nozzles.
So I think we can pick one of the nozzles or the temp probe to be on that spot.
Yeah, well, I mean, I've been in Alvin before where Hickey did it, and so we triggered one bottle at a time with rotating the tom thumb. But I don't know, it's sort of dicey, frankly. Yeah. We're at 180. I know, exactly, right. Well, I mean, it's one thing when you're doing it when you have some bottle that's recalcitrant, but in this case, the bottles are rigged fine. So Jill, we got up to about 180 here, and it's the same spot that we just took the other major, but it's not doing it well. It may be going up. Let's just give it a minute. It's 186 right now, but it doesn't seem to be. I don't think we're in the good flow right now.
Yeah, let's see if it goes up a bit more, but this will probably just be, it is what it is, right?
Yeah.
But this is why
the only
taking
other
so many samples. We'll have a good regression.
Yeah. Right. What you're saying, that one right there? Yeah. Well, that's what we thought up on the top, but the problem is it's so tiny, it's so narrow that we can't really get the nozzle in. So the only thing that I can suggest is you pull the bottle out, get the T-probe, and roto-rooter it a bit and see what you do, right? Because that's the only other option we got here. I mean, we know that it can be productive, but it's just clogged a bit. Frankly, if we're going to put the temperature probe in here anyway, we're going to need to make something that's better, a throat that we can stick in better. Oh, for the right. But you want to just move over a little bit to the right, sort of split the difference. Okay. I mean, this top one here seems like it's falling pretty well. Well, I think with a T-probe, you could try sticking the temperature probe down the throat of it, because it probably would fit, and then sort of wiggling it around and see what happens. Yeah. Yeah, it's the IPC cam that's super close to the. Oh, hello. I could see, yeah. No, you're off of it. It's just close. That should be some interesting footage. So this is a view looking down. I'm looking at the fisheye view from my dual minute GoPro, and it should be some interesting footage there because it's looking sort of straight down the structure. Take a snap of that. That's good. You can reach over far enough and put the claw down. It looks like you still have enough reach on the minute to do that. So it's this one here. It's right that. It's that. Yeah. It's up there. And it's, it's at that angle like that. Right. Well, frankly, I think, just getting the probe inside of it, and as I said, sort of wiggling it around to see if we can rotor it. Yeah, it's up there. It's over to your, right and back. We're almost there. What's that? No, you're in front of it. I'm looking at this camera right here. Yeah, there you go. Still in front. You're aligned with it, but then you just have to go straight back. Yeah, exactly. So now you're in back of it. That's looking pretty. Yeah, all good. That's close. Okay, so now what do we get for temperatures though? We're 101, 99, 86. Yeah, we're not really in it right now. We're just trying to line up. It's quite a narrow orifice. You can't zoom in on this guy on your pilot cam? Oh, I see. Oh, those are the flex links on the side. Open up the ISO, please, so he has more. Yeah, open it up more. That's it. Good. There you go. Yeah, there you go. I would just walk the probe back. It's that one. Yeah. That's where the flow is coming out of, right? Right there. Right there. That's it. That's it. They're in. Yeah, right there. And then to the left. 118. 108.97. 87. Come on. What the hell is going on here? Oh, you're out. No, absolutely. 76, 93, 109, 123, 135, 146, 173, 180, 86, 192, 208, 212, 217, 221, 225, 236, 241, 252, 260, 2, I thought it was going to get to 270, but it's not. It's 269. Go down in it anymore or not? 271. Will it go in or not? No? I can't tell if it's moving or not. Is it stuck? 267, 264, it's going down now. Oh boy, I don't know, Jill. I'm not sure that that's going to work. I mean, clearly there's reasonably hot fluid, but I don't know that we can. The nozzle, the major nozzle tips are going to be too big. You want to go back and try to excavate down at the first hole. Let's try that for a minute and see what we get. So that sampling spot right down there. So that's see what you can do to make a throat in that one. And it looks like it's coming in behind this little feature here, right? It's up against the wall. Right. Yeah. It looks better. Okay, I would do that again and see what you get. Let's shove the probe in and see what we get for temperatures there. No, it's not climbing very fast. Can you go back into it? We're about 111 right now. 126, 127, 126, 121. Hold on, he's coming out of it now. Hold on. Not sure. We're 112, 33. 160, 170, 180, 90, 190, 200. 215. We're sort of hovering around 220, 221. What did we get? We got 3 something, right?
Yeah, the max we've seen on the IGT was 335, and the
Right.
earlier maximum on the T probe was 324.
Right. So it's, I mean, it's still climbing a bit. We're at 333 right now, 334, 336.
Really?
Sorry. No, my mistake. No, 228. Sorry, 228, 227. Yeah. A little bit of sleep
Well, that was better
deprivation.
than what we were seeing on the second pair when that was in the fluid. So
So, right. So it's 230 right now. 234, 235. It's rising slowly. And I think this is big enough so that we could fit both nozzles in. It seems like it's stalling out here. Coming up again now, 246. 250. 254. 260. I mean, should we go, and now that we've sort of made a bigger orifice, should we just go get the bottle and see what we can get?
yeah, I think we should do exactly that.
Okay, all right, so let's take the probe out, stow it, get the bottle in there. I mean, wait, as long as you got it in there, let's just wiggle it around a little bit and see if we can make something that's, by the time we get the bottle in here, might be a little more. And you don't think you can go in anymore?
So I'm hitting it soft so I can penetrate deeper into it, but we don't want to do that with the majors, right?
No, that's what I'm saying, but we do want to do it with this. That's what I'm saying. If we're going to excavate, we got to excavate with this guy. So I would try to jam it in a little bit more and just wiggle and try to make it a bit bigger. Now you're out, right?
It's just I gotta kind of come out.
Yeah. Yeah, I mean, it's the hole right above this guy right here.
Able to poke that in pretty deep.
What's that? When it did just now? I'll do that.
Just be wary of, this friction as I'm doing stuff too, but back to that.
here. Well, it looks like you really need to be sort of coming in at it from there.
Straight.
Well, straight or angled that way into it, into the wall. Because you see, it looks like it's coming in through here and up the side, right? So the orifice is probably back in there. Yeah. Right. $89, $100, $145, $154, $164, $72, $89, $96, $200, $210, $230, $240, $250, $260, $270, $280, $290, $299, $300. Okay, I think that's the ticket. Okay, let's get the bottle in there before
Nice
it decides
job.
to do something different.
Yeah.
All right, it was up to 312, 313 before we took it out.
All right.
316.
Excellent.
Okay.
That's great.
All right, so let's try to just get it in there, Chris. Yeah, that's fine. Yeah, what'd you just hit? It could be the camera on the top. Okay, that's fine. Yeah. Well, the camera's still attached anyway, so that's good news. Yeah, I think you did some good with that. I think you got it. If you just swing the arm out, it's going to take it away. It's going to take the. Right. Yeah, I mean, the issue is you're jammed up against the chimney on the left. That's the problem. All right, you're going to go and get some good fluid from that smoker that's right in the middle there. Just turn down the ISO just a little bit. Or you want me to do it? Yeah. That's good. That's good. And I think, I mean, is it in that alignment, is it possible for you to come in so that you're more sort of shoulder down, so you're trying to get the probes back like that? Okay. What's that? Yeah, okay. Okay, we're trying to line up. We've got the ICL is working, and we're just trying to get the probe tips lined up against that back wall of the orifice that he excavated.
Probes are contacting the base there.
Mm-hmm. $39. I think, yeah, I think you're, yeah, I think you need to go there to the right and then back and into it like that. 79. That's 35 now. $77. 101, 103. 176. 214. 244. Okay, those are the kind of jumps I like to see. 258. Come on, baby. 268. 271. 70s. 277. 273. Come on. What is it now? 280? To get my glasses on. 282. 273. Oh, sorry. You want to go back and down? I think you can go down a little bit.
Yeah, so the problem is,
Yeah. What?
this is hitting here,
Yeah.
and I can't get it.
You can't get it that way, right?
So I can try to just kind of up and down and chop away at the.
Well, I don't know that. Well, right. But I think getting the nozzles a little bit further in would be good. I mean, just try going down a little bit and see where we get with the temperature. Oh, now that piece is going to fall in on us. 238. 266. Nope. 268. We're stuck around 260s, mid 260s, Joe. Is it giving us a reading or not? No, the ICL's not working. Oh, now it is 277. To the right, to the right. Give it a try. 284. Hold on a sec. 284.9. No, that's an old one. Okay, 276. Yeah, maybe try just a tiny bit, just a tiny jog.
Yeah.
275, 265. 249. Nope, that didn't do it. But try this. Try just going down slightly and then come back to the left slightly. You able to go down or not? No, that's it. No, now you're at 153. Ah, crap. No, we're down. We're back down to 127 now. So Jill, I think we need to figure out if we can get it back up to around 270. Is it worth taking the bottle or not? $68, $234, $234. $221. Okay, 114. 26. No, 68. $41. 100. 90. 185. 258. 284. 297. Okay, let's get ready to take this bottle, please. 299. Shit, what just happened there? 269. 268, 293. Okay, is Ronnie ready? 300. Hold on, not yet. Jill, if you're there, we're at 300, and I would vote to take the bottle. We're at 308.
Yeah, I'm back.
Okay.
Let's
Okay.
take it.
We're at 308. Let's do it now. Yep. Okay. Okay. Bottles are, oh fuck, we're moving to bottles. Rats. Okay. 280. Okay, so it's coming up. So looks like it's, yeah, exactly. But the issue is do we see that are we seeing flow out of the, oh, that's right. 287 as the springs are coming up. 288. So we're still in it, but the springs are still coming up. So just note that, Bill, in the record. We had gotten up to, I think, 308. And so it's 283 right now. Springs are still coming up. Yes. Yeah. We got a bunch from this already. Jill, do you want that? Do you want him to get that piece or not? We did get a piece of M-Vent when we came the other day. In fact, a giant piece, but
Yeah, I remember that piece.
Yeah.
If it's something we can grab, it would be good to have one that's in contact with this fluid.
With this fluid, right? Okay. All right.
Yeah.
So yes. So the answer is yes, Chris. Could be, yeah.
Although the other sample will also be very useful.
Yeah.
I'm very excited about that big one.
Right. Taking a couple of snaps of that. Yeah. Okay. Thanks, Rose. And the temperature is 292, and the springs are, I would say they're fully retracted. Chris, what do you think? Still coming up or not? Tiny bit. Yeah.
One of them's
Tiny
kind of
bit.
slow here. The left one seems slow.
Well, a little bit, but it's maybe the perspective. It looks like it's stopped. The one on the right looks like it's up, and it's now stopped. I don't think that, I'm just looking at the tip. It's not moving out towards the edge. You think so? All right, well, we can leave it go for a second. It's 293.8, 293.9 is what the temperature is reading. 294 now, 294, 290 something, 295. Okay, so we're at 295 here at the end of the syringing. Yeah, yeah. Right, the far one is still coming up a little bit. The far spring. And we're at 293 right now. Okay, we stopped. Good. Okay. All right. The bottle stopped. We're at 295.5, 295.7. Okay. All right. Let's go. Let's take this bottle out. And we got the lily gas tight to do. That's going to be fun. Although the nozzle's definitely is thin, but that was the one. We're not sure whether this was did you remember which lily bottle was the one that we could get good contact? Is it noted? What's that? Okay. There was one that we had fitted up the ICL that worked on deck, and the other one was not really aligned very well and didn't work, and we bailed on it. Okay. So in this record here, you basically got it as we're continuing to sample and which bottles are being sampled. Has that been noted? Okay. What's that? Yeah, right. Okay. Okay, I can just stay there right there. And the lily bottle. Okay, you see where that is, right? It's right. Yeah, yeah. You see the coil right there. And you're gonna have to fish it out a bit. Yeah, the handle is being blocked by these stupid markers. And we've got this M vent thing right here. So if you want, you can grab, why don't you grab that and let's put it out and get it the hell out of the way. That'll make it a little clearer to see what's going on. So you can grab that. And you can probably put it like right over there in the rubble over there. Can you, yeah.
So can I grab it?
Absolutely, yeah, yeah, yeah. Just don't crush it if possible. But yeah. Okay. Yeah. Yeah. Yeah, you can just shimmy down the thing and you'll hopefully, there you go. There you go. That's good enough. Yeah, just keep tugging. some shackles attached to the bottom. There's temperature probes. There's all sorts of crap in there. No, I think that actually was Zoe that put all that crap in there. Yeah. Yeah, the shackles are probably underneath everything. Yeah, that's good. You turn down the ISO. Perfect. Look at that. Come on, baby. There you go. There you go. All right. Good. That's good. Very good. Yeah. So put it in front of the bio marker there, up on the top in that in the rubble bits. Yeah. Yeah. Back there. Yeah. Why not? Very good. Hopefully it's not going to get in your way when you go to sample the lily bottle. Right? That's all we need. So the handle is down. Yes, that's right. Yeah. So yeah, so yeah, so my guess, so look at this one here. See? So my guess is you're probably better off just trying to grip in there. See the handle is in back here. The handle is resting on the corner of the bio box. So this cable here is all part of this marker, right? Yes, right. Yes, that's right. That's right. But down here is not too bad. Yeah, you can grip on the bottom part of the bottle by the, by the strong back. Yeah. No, I think that you're doing all right. There you go. You got it. No problem. Yeah. You're going to do a handoff? Yeah. No, no, no. I think you got to hold it and then give it to the other manip and then do a grip on it. You think it'll be reasonable to just rest it there and do it? Right. 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 Yes. It's got the ICL on it. Yep. Well, we thought it was a good idea to just leave it there. Of course. And you've got the same problem with my camera up there. But you're close, but I think you're still clear. It's definitely getting a bird's eye view of all of that stuff. Well, remember we fired number 4, so it's not going anywhere, so you don't have to pull it out. Like, yeah. Okay, very well. So I think probably the deal is you want to see if you can't put it up and have the other manip grip it so that you can get exactly right. Yeah, just be careful of the cables and connectors there. But I think next time we're just going to have to find a scabbard for it because, well, we didn't put it on this time because we thought it was going to make the basket that much more complicated, but this is a little bit complicated too. Excuse me. But after you get the sample, you're going to keep it in the grip and we're going to just drive to the elevator and dump all the stuff. Yeah, okay. Take a snap of that. You had to grip it below the plate? Is that the plan?
I'm gonna grip it around.
Oh, I see. Okay. You think the fingers are going to go in there? Yeah, the other ones will, the fatter ones. Yeah, that's it. There you go. Wait. No, don't let go.
Just grab it by the lower.
Yeah, okay. Yeah, that's a better grab. Take a snap of that.
Keep it down, please.
We're just entranced by the skill. That's what it is. Natalia, can you hand me my phone for a sec? Thank you. Thanks.
Answer to that question.
That looks like it's got it better. Take another snap. Well done, Chris. All right, meanwhile, I think the orifice has cleaned itself out a bit. Looks like it's all part of the plan, exactly. All right, we're gonna go and finish the water sampling here at MVent with a Lilia gas tight. Stop. Nope, 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 nope. That's it. Stop. Yeah, exactly. I think this may have been one that was not working very well in terms of communicating. So, right now, so you have to try to bump it out a little bit, best you can.
So if you look, it's touch.
Yeah.
We're gonna have to bump it out with it here.
Yes, exactly. No, I get it. So we'll give it a try.
Bearing in mind that if you hit that stopper, it's like real. I don't think we're gonna be able to get this.
It's all right. Just keep going. Anything? No? All right, let's position the sampler in the vent. Yeah, exactly.
Are there no comms on that one, Dan?
No, it's not, it's kind of wonky. It's not aligned very well. And we had a problem with one of them. We believe it's a screen one that it was, the alignment somehow was off. I'm not exactly sure. We'll have to check that when we get back on deck. So I don't know that we're getting, no, we're not getting comms on it. So I think we're gonna have to do the best we can. You're on the chimney structure with a nozzle. Don't go down. If anything, go up so we don't clog it. No, up. There we go. Okay. Can you zoom out a little bit with that? What's that? Oh, sorry. And remember, one of the things that Marv said we should need to keep in this mind is he said that we could bend the snorkel tips a little bit, that might help us in terms of that we bend his snorkel tips a little bit more. Okay. And be careful not to poke that down. Yeah, because those are really tough in terms of.
So that rock we talked about collecting, I think is going to get.
Yeah, that's all right. No, there we go. There's a couple of pieces there that we could still grab, I think. But let's deal with this guy first. Okay, what are you okay, but and you think that you're going to be able to do the plunger too?
Well, I think so we can.
I think
I think just the angle of that.
Yeah.
Makes me think that we're not going to get any from this.
Yeah, I hear you. So, but I think in terms of the flow, let's focus on the tip of the bottle and then try to just deal with the triggering.
This doesn't have an exhaust on it, right?
No, and it triggers sort of immediately. You press it and then retract it. So, but I'm thinking that the flow is behind your tip. Can you zoom in on that?
So I can't shoulder up anymore.
Okay.
So tricky to try to get.
Hey, Jill, it's a question for you. So my question is, do we go ahead and try to trigger this as best we can, or do we save it? I mean, I think that we can get it close to the flow. I mean, we're close to it right now.
Yeah, I think you guys can see better than I can visually.
Yes, yeah, for sure. We're looking at the pilot camera, and it's definitely better.
So I think if you think it's in the flow, let's take the sample.
Okay.
I think it's worth doing.
Okay. All right. So what do you think?
But
I would come down a little bit too, but be careful not to bury it.
Yeah, be careful not to bury it.
That's good.
And
Yes. Stop.
Be
Stop.
very sure it's in the flow. Sure as you can be.
Well, I think it's definitely there. You can't zoom in anymore on your camera. Yeah, it's definitely. The only thing I can suggest is, and yeah. So I think you should go ahead and do it. Yeah. Are we on the puck enough? Yeah. Yeah, we are. Keep going, keep going, keep going. That's good. Okay, and now retract. Okay. All right, Jill.
All right.
All right, let's go. Let's okay, now the one thing that we haven't done is install the temperature logger in here, but the one option that we have is laying that bottle back down in the box. If we can, that would be best, and then we can see about putting the logger and jamming it into the vent, and then we'd be done here, except for getting Rose's rocks. What's that? Where is it? It's in the manipulator. Great, perfect. Okay. What's that? The other lily worked, right? We're going to try to put it on the right side there. Yeah.
Do you have any preferences to which logger we grab?
No. Anyone you want. Yeah, that's good. You just shoved everybody over. Good. Okay, that's great. So you can just grab that logger on the right, the one with the red thing, and you want to grab it up by the tip. Yeah, where it's going over the edge. By the probe. No. I don't. No. Yeah, just grab it inside the box slightly. Halfway up from the red tape. Oh, fuck. Damn marker. Okay. That's good. All right. It looks like that's 2017 -007. So that needs to be written down, please, Natalia, in the C log. So we've grabbed the logger 2017 -007, and we're going to try to put it in the flow here. So this is going to be the next trick, although it'll be easier than the sampler because we actually are trying to jam it in, and it wants to be with a logger float going out that way, out into midwater. So take a grab of that, please. Right, and then from there you can just with the manip just try to drive it home into the vent. But definitely try to get it into that throat. If you can sort of angle it down like that. Take a 4K clip, please. You think you're in? Oh yeah, that's pretty good. Yeah, you don't think you can go any further, right? Yeah, okay, try it. Okay, that's good. Stop. Okay, can you back out a little bit? Yeah, can you just with a claw just push it over to the side so we can back out from it? I think it's still going to be okay in this direction. It's not up against the vent. And maybe push down on the bio box so that that's not complicating our life either. Yeah, yeah, and then just back out. Yeah, that would be good. And then let's see how it ends up hanging. Okay. You have the clip going, right? Okay. And can you try to get some, go down a little bit and try to take some pictures with the port manip? Okay, so 2017-007 installed an M vent. It's hanging out in the breeze there. That's a good placement. Let's get a grab of that, please. Yeah, thank you. That's great. Another still, thank you. Okay. You zoomed all the way out? Great, thanks. Okay, that looks good. All right, so
looks great, Dan.
Yeah, hi Thibaut.
Hey.
So that's we're at MVED, Thibaut.
Yeah, I'm relieved you succeed in placing the probe, because I was a bit concerned with those orifices so small.
I know. But the pilot did a great job because he grabbed it at the tip, which is key to this.
Yeah, great job, that's for sure.
South. It's south. It's the next one south. It's the big chimney to the south. So, but before you go, yeah, can you just with my camera, with a manip camera, try to get that? Yeah, that's good. That's good, Rose. Take another snap. Okay, just steady up on. Yes, that's okay. That's good like that. If you can. The money shot. Good. Stop. Fine. Yeah. Okay. This is at MVent. Good. Okay, you done with the clip? Thank you. All right. Okay, so let's. Okay, so it's 2:20, 2:30. So we got 4 hours left. So let's try to go get quickly get Rose's rocks stowed in the starboard side, and then we're going to go to the elevator to dump these samplers off and get the new ones and go to BioVent. Hey, Jill, if you're still here, the temperature at BioVent, do you have that noted? Or Thibaut?
From which year?
From 2019.
I will have to get in my folder. I'm actually streaming from my phone, so I will give you that in 10 minutes if Jill doesn't have it before.
Yeah, no, that's okay. And do we have it noted? Are you here all alone? What happened? Oh, all right. If we could go into the sea log to see where on our first dive on the 1306, and it was sort of at the end of the dive, what the temperature was at BioVent. At BioVent, yeah. Okay, we're just gonna go a little bit south to the other main structure here and look up on top for some rocks that Rose Jones, the University of Minnesota, is going to try to get.
Dan, would you like to return and look for stockwork on another dive?
Yes, because I think we're going to come back up here to deal with Q and that other little event that we saw, and so I think we're destined to come back up here. Turn the ISO down.
That sounds good.
That's good, Rose. Thanks. So are these the ones that you're looking for? The ones on the top? Are these? Well, these ones on the top are very easy for him to grab. With the one right above the anemone, and in fact, if you could get an anemone too, I'm sure somebody would appreciate it. Not me, but somebody would appreciate it. So pan down.
So what's our plan after we go to the elevator?
We're going to go to BioVent, which is about 100 meters north.
Where do you want the anemone?
Okay, yeah, in with her rocks, because they're living together and they're going to have to live together in the bio box. Starboard swing arm. Right. I mean, is that fair enough or not? Yeah. Okay.
Dan, I've got the BioVent temperature as 300.
Okay, 300. So not too much different than MVent.
Correct.
Right, you don't want to get that big one?
Oh no, I'm going to.
Oh, good. Yeah, there you go.
So bigger the better. Maybe.
So we're looking in the distance at the MVent structure where we place the probe. Yeah, and it's just south. It's the next chimney south of the one where that we instrumented and water sampled from.
Dan, I'm gonna go have a nap.
Yeah, Jill, thanks. So we're gonna be up at BioVent and get set up. You know, I would guess probably in hopefully an hour. We got 4 hours left before we have to leave the bottom.
Yep.
Okay.
Can you ask my watch standard to send me a message, and that'll get me up?
Billy, Jill wants you to send her a message when we're at BioVent. Okay. Just a text message, Jill?
Yeah, using the lab at sea Gmail.
Using the lab at CG email.
That's important, not WhatsApp.
Hold on. I'm going to pass you to Billy.
Thanks.
Hey, Jill, you just want me to send you a WhatsApp message or?
No, use the lab at sea Gmail account. Send me an email with that account, and it's going to play an alarm on my phone to wake me up.
Got it. Okay, I'll probably have to run down to get one of the computers. I'm not sure if the one up here, not sure if one up here is set up for it. Or I guess I'll try and log in the Zoom one.
Yeah, use the one that's running Zoom. Do you need the password?
Okay, sounds good.
Do you have the details to log in?
Yeah, let me just check. Yes, I got the info.
Great. Okay.
Okay. Yeah, I'll let you know.
Use that. If it's a WhatsApp, I won't hear it.
Okay, sounds good.
Thanks, Billy.
Yep, no problem.
Who is it? No audio. There is audio, but we're just doing a little survey here, looking with my down-looking camera, flying over these two vent structures in the M vent complex. There are a series of at least 3 chimneys, one inactive, probably the older one, and then at least 3 of them that are active in various states. Temperature that we got was above 300 C, and we're just doing a survey along these 2 northernmost ones. And then we're going to go to the elevator and switch out bottles, and then go to biovent for the last portion of the dive. We've got about 3.5 hours left before we have to surface. Okay, Chris, that's good. Hang it right. And just leave the down-looking camera on. So you're going to go up on the axial trough wall here, on the east wall. So you want me to come up? You got it. It's a little bit, yeah, you got another couple of meters for sure. And if you point my camera up and out, that's probably going to help you in terms of seeing where you're going. Yeah. And maybe shoulder down a little bit. So which way are you heading right now? You're heading southeast, right? So you're going to want to keep going in this direction, or maybe just a little bit to the left.
Yeah, so we do have it.
Yeah, good. Okay. And the tide gauge is right beside it, so. I still have a little bit over there, and I have to go pee. So if I drink more water, it's not a good idea. So we're flying at around 6 meters altitude. Yeah, this is the 2005-2006 flow, right along the east margin of the axial trough. Yes. Yeah. Hello. Now is it working or not? Hold on a second. That was Tim. Yeah, Tim. There is audio. We were busy doing a bunch of stuff, so yeah. So I was mostly focused on talking with the pilot about sampling strategies. The vent orifices were very small. We had to excavate one. We did manage to put the logger in the one that we sampled. So I think we got a reasonable set of fluid samples from it. And we're just going back to the elevator now to dump these bottles off and pick up the bottles that we're going to use to sample at biovent. So this is the east margin of the axial trough wall. Can you do a 4K grab? A 4K video snip? This is a great. That's good. Don't brighten it too much. Yeah, that's good. That's good. All these collapsed selvages. Oh, that's a beautiful archway. Take a snap of that, please. And there it is. There's the elevator. Wow. Can you just hold that? What a great shot. So there's a bunch of archways and lava pillars. It's going to go down. Yeah, yeah. Sorry, you're okay. Never mind. Keep going up. Keep going up. Sorry. That's that you can blame on Rose. That's Rose's rock. And the view on my camera is going to be fabulous. All right, let's keep going. Let's go to the elevator. Quite a nice view. Man, just gorgeous. Okay, so just to orient yourself, you're going to want to go to the other side, away from where the bottles are, the left side. So as you're facing the elevator and the checkerboard, you want to be on the left side. That's where you're going to dump the major samplers. Yes, so let's do those first. And the nozzles go over the side where the pink tie wrap telltales are. So the nozzles are going to be facing away from each other, and the springs are going to be down. So that's the, yeah. Well, to be honest, it shouldn't be too difficult. For instance, the major pair in the front, if you put that in the left side bin, you ought to be able to grab it pretty easily and then drop it in. Yeah. And you see where the tide gauge is. The tide gauge is just off to the right, to the south of you. Yeah, so I would suggest that we dump both of the major samplers, and then you can go get the two that are gonna replace it. And is a clip, the 4K clip done? Okay. Here, also hold on a second. I'll give Rose the microphone.
Hello, this is Rose Jones. I hear someone's asking about the rocks that I'm studying. What was the question? Okay, so the question was what I'm studying, which is I'm studying the progression of the minerals as they alter from very fresh rock to this very inactive sulfide rock. And we've been sampling the site and previous dives and got the active rocks. And this one was going back to retrieve more of this particularly interesting inactive rock because it has this sort of different structure and different microbiology and these different mineral structures. So, oh yeah, and I'm a postdoc at University of Minnesota with Barreyre.
For those of you unfamiliar with ROV ops, so the way that we're reballasting because of the rock that we took on is we're ditching a couple of steel plates. Each of them weighs about 16 pounds. You're going to get rid of one or two? Okay.
You're surprisingly neutral.
That is surprising. Okay, thanks. What's that? So again, the nozzles are going to go by the pink tie wrap telltales, and the springs are going to go down. There's a 2x4, actually, two 2x4s. So it's a 4x4 that the nozzle end is going to rest on, so the bottles are tilted down with the springs down.
So the temp ropes are going to be left and right.
The temp rope, yeah, the nozzles are going to be left and right, yeah. So that front bottle easily can go in the left container. That's an easy grab. The other one you'll probably have to reposition a bit. And I'd suggest putting the bungees that are on the inside on first on the handle, and then the outside bungee just goes over the nozzles. And you can turn my down camera off if it's still on. Got it. So the down camera's been turned off. Note that. At 1600 today. You got eight hours to deal with the fish suction system for LD.
We're calling that the AB Turbomax.
The what?
AB Turbomax.
AB Turbomax.
Ah. It's named the JA10, and that just does not flow. We feel like a thruster should flow.
Yeah, and if you can with the gripper or with the wrist, go like that, you know, and then shove the springs in down this. Yeah, that's the orientation. So I would note in the log, please, that we're putting the major bottles in the elevator. These were the ones sampled at MVent, and they are going to be put springs down so that we hopefully keep more of the fluid in the bottle. Excellent. Take a grab of that, please. Yep, there you go. And then put that right hand bungee over the handle. Yeah, baby. Pull it. It is tight. It needs to be. Come on, you're strong. Thank you very much. If I could do it, your manipulator could do it. How's that? Okay, and then the other ones. No, 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 you're underneath. Okay. Okay, that's good. And then maybe can you knock the bottle back to the left? Yeah, just leave it on the top there. No, over the edge to the left. Keep going, keep going, keep going, keep going, keep going.
Over the curve?
Yeah, right there. Okay. And then with a manip, shove the bottles down a little bit. Yeah. Just reposition it. Sorry, it was. Oh, it was Rachel. Oh, on the iPhone. Okay. She's up late. Does that look like it's stable in there? Okay, that's good. All right, good. Next. Okay. What's that? Oh, sorry. Take a snap of that, Rose. I think it needs to be back a little further. Can you shove it back a little further? Otherwise, I don't think it's going to sit properly. That way? Oh, is that right? Okay. Okay. All right. Let's just go with that. Yeah, put that bungee over the handle. This top perspective, I'm not. No, it's sitting fine. Yeah. There you go. That's one. Good. And the other one. Yeah, I think that's probably a smart move.